drop down towards the bottom.
Okay.
This is coming into view now, and it looks like we're getting sort of a boulder cobble sediment mixture.
Got a good visual on bottom as well.
On the substrate right now, and looks like we've even got a little sponge over here right off the bat on the
I'll
left
acquire
hand side.
bottom, and then you'll just get your 15 or so.
Yep.
Delta, and we'll get tuned in. Workshop pilot. That might be an issue. You might want to speak with Dan and try to get a hold of engineering, or speak with the bridge and try to get a hold of engineering, and make sure that that is actually that we actually have saltwater cooling piped towards us and running. Thanks.
Were we pretty hot on winch?
Yeah, sound like it. I don't know if it's normal or not, but I'm gonna take a look and make sure that everything is okay.
For today's dive, it's sort of a ridge or a little promontory that's sticking off the side of a ridge. It's fairly steep, and we are starting in one of the steeper parts. And as I recall from the backscatter last night, it was sort of moderately reflective, and that seems to be what we're seeing right now. And as we continue upslope, I think the reflectivity is going to increase.
I've never seen it above 90.
And therefore, we're expecting to see perhaps more consolidated substrate about halfway.
Bridge, this is Nav.
Okay, spin this up a little more.
Go ahead. We just hit bottom. I'll update you on the bailout as soon as we start moving, okay?
I'm sorry, can you repeat?
We just hit bottom, and then I will update you on the bailout plan as soon as we have it.
Understood, thank you.
Yes, Chris, I think it looks like a
field of rocks. 10, so I'm going to drop some more.
Okay, I'll come down with you.
Probably shedding from a little ways above.
I'm just trying to get my footballs in view.
Sort of near the top of the dive, the
there we go.
topography
Look good.
Seems to sort of be less steep. Maybe there's some sediment up top that's sort of shedding down and filling in the gaps between the rocks here.
Right, so it's fairly barren of animals so far. We've got what appears to be a small coral that's going to be coming into view, and we saw a sponge earlier, a stock sponge. And so as we come across animals as we go upslope, we'll take a peek and see what they look like.
So Chris and Jasper, do you think all of these rocks are in place here? Do you think they're sort of, concreted to the seafloor, like this is the seafloor basically?
Okay.
I would say probably a number of them have rolled. These don't look necessarily in place, all of them. They sort of look broken up a little bit, and so usually you get, when you get pillow lavas, these things break up and parts start rolling downhill. So some of the bigger ones may be in place, but the smaller pieces may well have rolled some way or a larger way. That's hard to tell.
So is there any interest in the stock sponge or the rocks here? Should we just keep on going?
If we drop down on a site where it looks like there's good rock collection possibilities here, which it seems to be here, it looks like there's a fair number of loose angular rocks. I would suggest that perhaps we start by grabbing a rock here. What do you think, Jasper?
Yeah, I think this is this looks promising. I mean, these could very well be pillow fragments with probably a good bit of manganese on them.
Can we get lasers on so we can see what we got for sizes
super thickly coated? So this seems like a promising spot to find a nice.
Also,
nice
how do you feel about deploying the light bars?
Our uppers are. We could do one, but one of them is currently sticking a little bit.
ROV pilot, watch lead.
Watch lead, ROV pilot.
Roger, Jeff. So, what do you think? Are we going to be able to perhaps grab one of these rocks in front of us, a nice angular one?
I can certainly poke around and see what's loose. Is there anything that jumps
Jeff,
out at you?
Your
I'm going to let
port
Jasper
motor
guide you in on
zero.
the collection here.
Oh, that's
GUI problem or actual problem?
Go ahead, Nav.
Yes.
Ah.
There we go. Ooh, that's gonna.
Go ahead, watch lead.
Oh, I saw you blinking. I thought you were trying to hail us
That's
here.
why I was.
Sorry.
Sorry
I
for
couldn't
that.
figure out what I was doing.
Yeah.
I was like, this is wobblier than I remember. Okay, so is there one? Sorry, there was a little bit of a quick fix I had to make. Is there one of you I didn't quite catch? If there's one of you here that you wanted to poke at to see if there's anyone in particular that looked good. Watch lead.
I'm looking around a little bit. I'm still having a hard time with the scale here.
Okay, so the scaling lasers are on, and they're about 10 centimeters. They're 2 red dots, almost center screen. So I can put that on one of the rocks, and you'll see this rock is roughly 10 to 12 centimeters. That one's width is 10 centimeters. This big guy over here, this would be too big to pick up. Looks angular. That's what I imagine the type we're looking for, but that's a little large. Would that be about right? Is this the sort of shape you're looking for, but smaller? Yeah. Yep. Okay. All right. We'll sidle over this way. I saw some angular things over here. See if there's any small ones or more reasonable sized ones. Doesn't have to be tiny. But like maybe this guy looks pretty angular right here.
Yeah, that
The
one right
lasers
below
are
the
just
lasers.
above.
Yeah.
Is that pretty big, or is that all right? Let's see.
Looks pretty
That's
good to me.
20 centimeters
Don't you
wide.
think, Pilot? That looks pretty good.
Let's get close and see what we can do with it. How's that look? Is that a reasonable
Yeah,
grab?
that big one that you just were on looked okay, yeah.
Okay, let me get close here and get myself situated for a quick snag of a rock. So I'll reach out, see if it's loose. Once we know it's loose, I will open up the rock box and stow it away.
So one thing, just looking at these rocks, and correct me if I'm wrong on this, Jasper, but I don't see the real heavy manganese incrustation that you would typically find on a very, very old seamount. Maybe these are because they're fragments that have broken off at some point.
Okay, you want to get spun up on hydraulics?
Can we assume that
you are
this is
medical and hydraulics.
Cretaceous seamount, maybe 80 to 100 million years old?
Well, let me guess, I guess, right? It looks like if you look on the bottom of the right screen of the screen, you see some of the more bulby-looking cauliflower type manganese crust. So I think it could be fairly thick. The particular pieces we're sort of aiming for in the middle of the screen, it might be a little thinner. Telling just from this view whether this is a Cretaceous seamount is a bit of a guess.
Yeah. Yeah, it's more just its general location on the Pacific plate,
Right.
and which many of these seamounts over in this part of the ocean are actually known to be quite old, just due to the movement of the Pacific plate to
Right.
the northwest.
Well, and there were some samples
It
in
wiggled
the
a very small
80s
amount, but
that
it
have
seems to
been
be ever so slightly wedged.
age dated at about 100 million
from a
years.
different
That came
angle.
from another
Yeah,
side of
it
this.
is kind of
this
wedged in there.
seamount.
I
So
see it move every time I touch it.
in
But
general, I think
is there another one in this view before I take
we're,
off? This guy's kind of wedged, it looks like.
we're pretty good. Let
Um,
me see. A different rock.
yeah,
But over to the left a little bit,
this,
maybe?
this
Yeah,
guy
maybe.
over here, maybe.
Maybe, yeah, that one.
Yeah.
Okay,
Yeah, that one maybe.
give this a little push. Let's see.
There you go. That's
That
it.
guy's loose. Is that one an okay sample?
What do you think, Jasper?
Would that be reasonable?
It's hard to see if it's flat and just manganese or if it's
Looks sort of rounded
a
to me.
bit more rounded.
Um,
Let's take a look.
I can give it a good squeeze too, and manganese will just crumble usually versus a solid rock. But we'll try first a lighter grasp, and when you say you want to try crushing, we will try crushing. Do we want to just put this in the starboard box since we're not sure we're going to take it?
Yeah, we can do that.
Okay. Rather than going through the whole opening process just to find out we're not going to pick it up.
Okay, after you pick it up, I'll wing in for you.
Thanks. Okay, got a grip with just the fingertips here. And we'll get it up in view.
It's got some girth to it, right?
Yeah.
We'll tilt up a touch with the main HD.
There we
Get it in the light pool.
Yeah.
What do you think?
Would you like me to come at it all? With the drawer?
What's that?
Do you want me to come out with the drawer at all?
No, that's okay.
See, now that looks
What's
a little bit, from that angle, it looks more like manganese.
Yeah, but is it thick enough where we
Yeah.
could have a core of basalt maybe?
Do you want to zoom on that pilot?
Yeah, why don't we
Well-
take a little quick zoom just so they can get an idea what they've got here.
Yeah, it's definitely got manganese crust on
Yeah.
it. You can see this very bubbly type of texture.
Yeah, exactly. And on the right, it looks a little bit more layered.
So what the decision here is whether or not there's actually basalt, the original rock
Inside of it.
it that's now coated by the manganese crust,
Right.
because we're far more interested in the basalt inside to be able to age the Seamount.
Right.
Although there are other groups that are very, very interested in the manganese crust,
Take a little
because
zoom
this
if
is
you
one
want
of
here.
the
I don't know
International
if
Seabed
it's any
Authority lease
different.
blocks
Looks the same bubbly.
here. But our purpose here is to actually try to age different seamounts throughout the Western Pacific.
So what's the call?
So, what do you think? I mean, we're going to get another rock later on in the cruise. I'll just go ahead and keep this one.
Yeah, I think we can. Why don't we just back it up for safekeeping?
Okay, we will keep it then, and we
Great.
will put it in the starboard box.
Okay, so we need to go ahead and start the
Let's tilt down so we can get a view of the box. People can see it go home. Yeah, we're going in the starboard box, if that's okay. Especially if it's going to be your least favorite rock, then. Want to make sure the good one goes home in the safest box.
manganese crusted volcanic rock, I guess would be the way to describe it. And not yet, yeah, he's dropped it. Perfect, okay.
Okay. Go ahead and
then,
close the
click this one back,
box.
and
up.
you're ready for the next one. So I did a bit of the procedure last year. Jasper's new to it, and so he's learning, and I'm trying to remember because I'm a little bit older. So
So bear with us.
Yeah, bear with us as we fumble a little bit on this first dive, but we'll get better.
We didn't.
Your call, dude. You want to do it? We can do it. Okay. No, not on the first dive. Okay.
Yeah, should we do some introductions here? With
Let's do it.
are we changing? We're doing an ROV change out. Got it. Okay. We'll wait till everybody gets seated, and then we'll.
Okay, so we just got you that rock sitting in the pilot seat representing the Global Foundation for Ocean Exploration is Jeffrey Lanning. On my right is my co-pilot, Andy Lister. On my left, the navigator, Fernando Aragon, sorry. And we are here for another minute and a half, and the next lead will introduce themselves when they show up. we go. Oh, they said you guys would do that.
It gets kind of passed down them. You're like, oh, and then I'll let my video introduce themselves.
Sorry.
Yeah, so as we're waiting and they're getting settled, again, you're watching a live broadcast from the NOAA ship Okeanos Explorer. My name is Chris Kelly. I'm at the University of Hawaii in Manoa in Honolulu, and I'm serving as a biology lead. And to my right
is Jasper Conter. I'm also at the University of Hawaii, and I am serving as the geologist on this cruise, and this dive, obviously. We just collected our first rock sample, or I guess the first sample of this this leg of the 1606 cruise. And we are doing a quick swap out of the ROV crew here. So we're looking around and pondering over other bigger, better rocks, but we're going to move on in a minute anyway, because these are not going to fit in our box.
So a couple things about the ROV, the Deep Discoverer, which is our ROV that's deployed off of this ship.
All right.
It's what's
Are
referred
we
to as a two-body ROV.
underway right now, or is the ship
And
stable?
it's got a,
We're stationary
it's
right
basically
Okay.
Tethered to the ship
I see
via
a sponge up
a
ahead.
main cable coming down to a vehicle that's called the Sirius vehicle.
We're gonna be
and
moving,
it's essentially a camera
at
sled. And
roughly
then a
330,
lateral tether
and we
comes
do have
off of the
800
Sirius
meters.
and goes
Okay.
to the
So
actual
330
Discoverer
is-
ROV itself, and that's if you're
slope.
looking at HD
Well,
camera
let's
Line
1.
up the slope a little better
That's
here.
the footage from that particular vehicle.
Yeah,
If you're
maybe more like
interested in
340
seeing the
initially.
camera
We'll
sled
change that
footage,
as
you
we
can
get
go
up
ahead and
further.
take a look at that by switching
I mean, you
the
look
feed
at the-
too. So we have a sponge coming into view, and I wonder if we could take a peek at this
Yeah,
before
we
we
certainly will.
start
Why don't
hitting
you,
up slope.
Find a heading that you're comfortable with, co-pilot,
Copy that.
and I'll take a look at this sponge, and then we'll get it underway.
I think this is probably the stock sponge that we saw as we were coming down. And I think I know what it is, but I'll wait a bit till we get a little closer view.
Yep, pilot.
Yep.
I'd say localize a little more towards northbound. I don't know how that correlates with high pack.
Yeah, I mean, it's
Yeah.
probably fine.
So this
All right,
is
zoom in.
a sponge in the class Hexactinellida, or it's a
Hey, Roland,
glass
let's go ahead and zoom
sponge.
in real quick. Just partial is fine.
They're called glass sponges
And hold there.
because they have a very glassy and sometimes
Maybe iris down a
translucent
bit.
appearance. This particular sponge is in the family Euplectellidae and
It's,
the genus Sacocalix.
6 hours, 30
And
minutes.
That's
so
a
some of
partial
the
zoom
sponges
pilot?
down
Okay.
here in deep
I'd go
water
in and
actually
call
have
in-
stalks,
go ahead and
as
call
you
in
can
the
see
move.
at the very bottom
Yeah,
of
not
it.
a lot of leash left.
And
Yeah.
then the actual body of the sponge
Okay.
is on the
And,
top of the stalk, and that's what
how-
we're
how
zoomed in
far
on
do you
right
think we
now.
Have they-
So
I would
this is
say
a fairly
30, but,
common
They haven't moved.
genus.
What have they been
yeah,
moving?
we've seen
They
in the
haven't.
Marianas.
haven't?
We've
Okay.
seen
Just
as
got-
well on Johnston
Right.
and off of
Well,
Northwest
up the
Hawaiian
slope
Islands.
would
So
be
it's good to actually
about
document it at
340.
this
We could do
location
30
as well.
meters.
Yeah, let's just do that.
Great.
Sound good to you, co-pilot?
Yep.
Okay.
Lose the lasers.
You can kill the lasers. All right. Chris, are you happy with this?
Yep.
You want to move on?
Yeah, let's go ahead and
Okay.
move on. See if we can get into,
This depth, move at 0.2.
more interesting
Yeah, point
hunting
two.
grounds
Great.
for biology.
All right. Let's zoom back out, Roland. You can keep the skids out of view if you want, if they're in the way.
And you can bring the lasers back on as you want. Holothorian on the rock.
see him.
The one in the left lower.
Bridge,
left
ROV
corner.
nav.
It's
Oh,
not that
there's-
I'm asking for it,
Yeah.
just
Sure. No, I
pointing
didn't
it
see
out.
Chris, any
Go ahead.
interest in a Holothurian?
We'd like a ship
Um,
move, please. 30
I don't
meters,
think so.
bearing
Let's go ahead and
340
move on
degrees,
on this
0.2
again, and
knots.
I
Okay.
think we'll find a little bit more variety, a little upslope.
Ship move, range 30 meters, bearing
Looks like
340 at
the
0.2
from this
knots.
view looks
Okay,
like
copy,
it's
thank
probably
you.
a synallactid.
So 340, you're lined up. I'm lined up. Looks good.
There's a swimming
Oh,
shrimp.
yeah.
Something interesting, a shrimp. Looks
Would you like to
it
zoom in on that?
Sure, let's go ahead and get a zoom. He's
Uh,
coming right at
You
us.
can go ahead and go partial.
And we don't want
and
to
hold
offend the
there.
shrimp since he's coming to greet us. So.
That's right. We come here to say hi, not to do any harm.
Nav, bridge, move initiated.
Thank you, Bridge. Seems to be something wrong with the time offsets on my calculator spreadsheet, but coming up at 4:30 is in
So I
6
think-
hours.
Yes, he
30
does
minutes.
have
Yes.
a
can go
rostrum, a fairly long
more zoom
rostrum
if you want.
sticking off
Hold
his
there.
head. And with these swimmerettes, these swimming appendages that are coming off his body, I'll take a stab at an ID. I'm not really great on these arthropods, but I think this is in the family Aristeidae, and it could be Aristeus.
Go ahead, survey one. Yes, that's correct.
So I think in the Eastern Pacific, Chris, we
Yeah,
saw
zoom out.
ones like this that had fairly long rostrums, and we were calling them Plesiopeneus armatus. I mean, they might not be the same species,
Just
but
going to keep
that's what we had
Going
some
lateral.
specimen in Central
Gotta
America.
know when,
I'm not so sure ID do that. I'm sure this would be a
Yeah,
totally
the
different
shrimp
look like you're moving.
genus,
But,
but
yeah,
yeah,
shouldn't take too long. 3 minutes.
Yeah, so that was the voice of Diva Amon. She's one of the deep water specialists here who's participating on this dive. Thanks very much, Diva. I'm trying to remember whether Plesiopeneus is in the Aristeidae. Is that correct? Or
It
am
says
I,
below us.
maybe I'm remembering wrong.
I am looking that up right now on Worms. Give me a
I'm going to
second.
do a snap zoom real quick.
Sure.
I'm just curious what this is. Any interest in this? Oh, that's nothing. Never mind. I saw a little speck of something. Let's come out.
Yeah, so it is in the family Aristeidae. So I think we can definitely say it's from that family, just as you said. But genus-wise, I'm sure Mary can help fill us in a bit more.
It looks
Sure.
like you're underway, huh, Copilot?
Yeah, and Mary will probably
possibly,
comment on this.
yeah.
This is Mary Wickstein down at Texas A&M who
Can we,
does specialize in these
Let's
animals.
range in on the sonar
And,
on D2.
Sure.
She'll no doubt comment by email, and sometimes she gets on the audio as
Do you
well
want 10
and comments
meters or is
on
20 good?
the
20 is
identification
good.
of these.
That's 20.
One of the things you look for on shrimps are whether or not they have a sort of a long spine coming off
Folder
the top
here.
of their head. It's called a rostrum. So there are certain
Maybe a sponge on
families
top.
that don't have that or have a sort of a reduced spine, and others that have sort of a longer spine. That's one of the things we were looking at on that shrimp and helped us narrow it down to an Aristeid. There's another one of these Secocalic sponges. It's like a little bit of coral. Yeah, we're getting a few more corals and things right now.
See a few creatures on top of this rock here.
Yeah. Maybe we could zoom in on that lyrate coral that's right in front of us. Yeah, this is definitely looking more promising, and you'll notice that the substrate's more consolidated, which is a pattern that we're seeing over and over. We're getting more animal life
Okay, let's
where
go
we're getting
partial.
more solid rock.
Hold there. And go zoom in more. Hold.
So this particular coral is in the family Primnoidae, a very, very common family in these type of waters. And I'm going to take a stab at this and sort of tentatively call it Calyptrophora angularis because it certainly looks like that. And as you'll notice all over the branches, it's got a lot of
You want to
brittle
go in a little
stars on
tighter?
it.
Hold
So,
there.
this sort of sinks home the message that a lot of these corals actually serve as substrate and are very important to animals that need to get off the bottom in order to survive. These particular brittle stars are probably filtering the water, pulling small things out of the water column as they drift by.
If you want lasers, let me know.
There's a starfish eating
We've
it
also
down
got
at the
some
bottom of the
anemones
screen.
on this.
Yeah, that's cool.
And I think they're
You wanna zoom
anemones
in?
or zoanthids.
Hold there.
And even a little, looks like a sea star, but we're seeing the underside of the sea star. So it's a little bit hard to identify.
It's a good catch, Roland. And what would you need to see to identify it, Chris?
For me, probably not much. Chris Mom, if he calls in and he's watching, he could probably tell us what it is just from this view. But the fact that it's on the coral, it's probably eating this coral. That's why it's on there. That'll narrow down the candidates for it right away, because a lot of the sea stars do not predate on corals. So that's your first clue right there.
Cool. Let's zoom out. Okay, are you fine with that view?
Yes, I am.
Okay.
Thanks, pilot.
We're moving on. All right.
Yeah, I'd say I'm halfway through the move, maybe.
Okay.
Not even.
So this is definitely part of the terrain that is much more rocky, much more solid than what we saw where we just landed effectively.
So what's the average slope of this
looks much more in
area
place
here
than,
in front of you
than what we saw below, which may have been pieces that came down
Right in
from
front of
over
D2
here.
or up the...
right in front of D2, yeah.
You mean from the multi-beam?
Yeah, from high pack.
Okay, yep. It's about 40 degrees.
Okay. So we just had a shift change in the front row. Thought we'd just introduce ourselves real quick. We've got Dave Casagrande as our navigator. I'm Dan Rogers as your pilot. And we've got Levi Unuma as your co-pilot. And Roland Bryan on the video station. And Art Howard back in the clipping chair. And we're all with the Global Foundation for Ocean Exploration.
Got a couple other people in the room here too. We've got Emily. I've forgotten your last name. I'm sorry,
Pilot,
Emily.
why don't I bring your sonar into 50 meters? I'm not seeing much out.
Yeah,
You're 80.
okay. Emily Narrow, and we've got Brian Kennedy over here to
yeah,
my
bring it
left
in
as
one
well.
notch.
Yeah.
And so
what I
we've
was
got
gonna
a
do.
full control room this morning,
Yes. Thank you.
helping
Maybe I'll play with your
to
gain a little too.
Okay.
guide this particular dive. Okay, we've got some more of these corals. Looked like at least two of them might be
How's
the
that
same.
move? Should we get another one going?
Looks like
Love
it
that.
just finished.
Co-pilot, what do you think?
So I'm going to swing a couple more meters yet.
Okay.
Give it a couple minutes.
All right, we can do that. Any interest in zooming in here, Chris?
Perhaps the sponge, maybe. I think the corals are all the same.
Okay.
I'm not absolutely positive.
I had a
but that
feeling you'd
sponge
want to
really-
watch or get a closer look at the sponge.
Yeah.
Zoom in. Hold there. Zoom in. And hold.
Alrighty, so I'm pretty sure that this is another glass sponge, and I think it's in the genus Tritopleura. At least I'm going to go with it. It looks like one, so oftentimes we'll ID an animal and we consider it more of a morphotype rather than a really solid ID, and it's more descriptive of what we think it looks like, and so on. That's definitely this case.
Yeah, I'd be okay with another move.
Okay, let's zoom out.
Yeah, thank you, pilot.
All right, we'll move on. And I'm going to come up a bit. So if you want to come up with me,
Yeah.
I'll face the slope here.
I think the next move might be like 350.
Yeah, let me come up this wall a little bit before we get that move
Okay.
in. See if there's anything interesting here that they might want to look at. Well, our tether cam is on D2 is flickering in and out.
Yeah.
Have you guys noticed that before
It's-
last cruise?
It's
I have
been
not.
it's
I
been
didn't
doing
notice
it
that.
during this dive.
Okay.
Yeah, it's
Jeff
not
noticed
last
it.
cruise.
Okay. Is it in the co-pilot log?
Let me check.
Well, any interest in anything here, Jasper? Any rocks that you might want to look at? I think you're muted.
These are all pretty nice and big and large and angular, but they might be a size bigger than what we had been looked at earlier.
So this is one of the long-legged shrimps down below in the genus Nematocarcinus. We can go ahead and
come up a few meters.
note one of those was here at the site. And there we go. Now we've got another sponge here, and this is in the genus Poliopogon. This genus is fairly common down here in deep water at these depths.
Can zoom in and hold there.
So again, it's a glass sponge, and these are very, very interesting sponges. I tend to be somebody who really likes sponges. And some people might go sort of, why? It's just sitting there. It's a big lump. But they are really fascinating animals when you really get to know them. And when you look at them microscopically, they have amazing architecture. Their skeletal components are rather extraordinary. One of the things we always note on this type of sponge is the way it's attached to the rock. And it's attached to the rock via very fibrous attachment, as you can see here. And that actually is quite informative. It helps us eliminate quite a lot of candidates for what the sponge could be. And I'm sure we'll see later on some sponges that have a different type of attachment.
You rolling?
Oh, yeah, there are.
More creatures up here.
There's a large one, and the rock behind this one, there's something relatively big.
Yeah, it's another Poliopogon again. And maybe one of these unbranched corals that's up above that. These could be bamboo, I'm not really sure. It would be good to perhaps confirm what type of corals these are.
Okay, sponge or coral then?
Maybe the coral. The sponge, I know what genus that one is in.
Okay, which stock would you like to look at? I'm
How
looking
about
at this
the
bigger
long
one.
Yeah, the bigger one,
okay.
that would be good.
Okay, you want to zoom in, Roland? Just partial, I'll move in a bit. There's something in the port rail cannon on that rock too, we could look at.
Oh, it's definitely a bamboo coral. I can see the nodes, and you will see the nodes too in just a few minutes when they, or a few seconds when they zoom in on it.
And you can go in tighter.
Got a couple of people that typically participate on these dives, and they specialize in this group of corals, and they can actually identify what genus these things are in, or at least what clade, if you will. It's a genetic term, by closely looking at them. And one of the things I'm looking at right now is if you look between the tentacles of each of the polyps, you'll see these spines. And that's informative, so I can tentatively perhaps call this in the genus Lepidicis, just again as more of a descriptive identification rather than a solid ID.
I'm going to go in a little tighter. Hold there.
So all of these polyps are coming off of one side, so I'm not sure about that ID, but we'll get some pictures and we'll show them to the two guys who really work on these species. That's Les Watling in the University of Hawaii and Scott France down in University of Louisiana at Lafayette. Also Abby Laponte and
Like
See
to
who
get
else
that
works
move
on
in whenever
bamboos.
we're able.
Abby is the other person.
Yeah, I'm ready.
Who's with Les's lab.
Yeah, you can go ahead.
Works on them.
340 is still fine.
340. Okay.
I'll move back into your center
Do
screen.
another 30 meters.
What is this thing
Yeah,
here?
so
You want to
there's
snap zoom on that?
Yeah, it's a dead sponge.
Oh, it's a dead
that's laying
sponge.
on the seafloor. I think it's in the genus Spidoscopulia. And it's a
Bridge,
glass
RV nav.
sponge. When these sponges die, they typically
Go ahead.
turn brown like
We'd
this.
like to
I
repeat
think
the
they
last
collect
move,
a lot of
30
sediment.
meters, bearing 340 degrees,
And,
0.2 knots.
Repeat last move. 30 meters, bearing 340 at 0.2 knots.
Identification,
Copy.
but you
Should
can
like
clearly
lasers?
see that it's dead.
Sure.
These live glass sponges will have that white, translucent appearance.
Okay, zoom out.
Nav bridge move initiated.
Thank you.
You like your swing arms where you're at, or do you want me to get the
Well, we can't bring the port up or out right now, all the way.
lowers?
But looks okay to me. Roland, are you happy with the lighting?
It's a little hot in the center. I'd like more spread out light.
Yeah, you could bring him out, slightly, 20 degrees or something. Just the lowers.
Yeah.
Any interest here, Chris?
Yeah, I'm looking at it right now. Maybe a snap zoom, I guess. I think it's just a
Okay.
smaller version of what we've seen, but
Snap in.
I'm not positive.
Hold there. What is up with the colors on that tether cam?
Yeah, I think it's the same thing
It's
that
really flaking
Calyptrophora
out.
angularis on this one. Definitely it's a primnoid in the family Primnoidae.
Zoom in tighter. What is it?
Yeah, that's a primnoid coral, so yeah, that's what I'm going to go with.
Potential dandelion or something in the background on
A brittle star?
Yeah, brittle
To the
star,
left
and
of the
there's
brittle
a little,
star.
what's
Oh, I
that
see
in the
what you're
back
saying.
there? Is that a sea cucumber maybe?
Something in the
Over
Oh,
crack
and there's a
there.
polychaete
in the
maybe
crack.
on the upper right. You can zoom
Or
in
maybe
here.
a polychaete, yeah. This is a brittle star. This
Something waving around back there.
I used to call all of this sort of type of brittle star Ophiocanthidie, but now I know better, and now I keep my mouth shut on brittle stars because I can't figure them out. That is a
It's
Diva Amon has said it's a polynoid, which is a polychaete worm, scale worm.
okay, let's zoom out. I was like, what is that bumping? But it's your swing arm. So the move is underway, correct?
Yes, it is. About halfway.
It's like a lot of crusting here, Jasper.
Yeah, this looks pretty solid.
HPU secure.
We just saw some nice big rounded shapes, probably
Sponge up ahead.
more larger pillow-like structures. But this is sort of the top completely covered in manganese. We're now coming up on some smaller, broken up pieces again.
Not a whole lot of leash left.
Okay.
Looks like there's plenty of there
If
were a
that,
number of folks
camera
liking
is
to
more
live up here.
distracting than it is
Looks a
useful.
little
I can
more
put something
barren
else there.
here.
No, it's fine.
There's
It
something in the middle there, maybe.
bothers me that it's not working, but
Right.
it's not
There's a good
bothering
sized sponge,
operations
maybe
at all.
a little bit
Oh,
further
yes,
up
yes,
on the
yes, I
other
see
on
it.
one of the other cameras.
Yeah.
We're able to see it.
Sorry, Chris, what were you saying?
Oh, I was just noting in one of the other cameras that there's a sponge that'll be coming up. in
Yeah,
view.
we're the ship's underway, and I'm just looking at
Yeah.
stuff in front of us before we have enough tether to move up there.
Yeah, sure. So, when we're talking about manganese here, Jasper, what is manganese crust?
So the manganese crust is it's essentially a precipitate from the ocean, and it's loaded with basically the transition metals. That's the metals in what people call the periodic table. Things like iron, manganese, cobalt, a number of these
Bit
metals
of a port to starboard current.
that are
Not too
commonly
bad, but
used in
definitely
various
feeling
types
it.
of industries.
Okay.
But they precipitate out from the ocean water on these rocks down at these great depths, and they form these thick layers on
fish.
top of these rocks.
Zoom fish.
We're coming up on. Oh yes.
Hold there.
Is that
Zoom
a
in.
fish?
Hold.
Sure is. We're going to wait for Bruce Mundy, who's on the line here, to tell us what this is once he gets a good look at it.
I haven't seen a good view yet, but this is a cusk eel in the family Ophidiidae. And this looks like something that we saw in Hawaii, in the Hawaiian Islands, that really puzzled us because there wasn't agreement among the experts
Hold.
on to study this family of fishes, the taxonomists, on what
Zoom
this
out.
was. I think we were guessing.
Okay.
I'm talking here, but I haven't gotten a really good look at it yet. So the fish isn't cooperating.
I'm trying to remember, Bruce, what that one in Hawaii, was that Kumba? Was that the genus for that you guys thought it was?
Okay, well, Chris, do you think that was an aclurid or an ophidia, a cusk eel or a rat tail?
You're right, sorry. that wouldn't be, Kumba's not right. Sorry, senior moment. You're right, I think it's an ophidia.
Yeah, so the best guess that we had was a genus called.
Okay, you want to zoom in on the sponge?
Okay, now
hold
I've got
there.
with my light on, I've got the view a little better. Let me take a look here.
And while Bruce is pondering that, we're also looking at a sponge right now, and it's in the family Eurydidae, I'm pretty sure. It's a very brittle sponge, and it's a hard sponge. So, but
Zoom
go
in
ahead,
tighter.
Bruce. You can tell us about that fish.
Hold there.
Well, just very quickly, we were guessing it could be in the genus Porogatus, which has short-snouted and long-snouted species. This would be one of the
Can we clear the lasers?
Turn lasers
larger
off.
scales in some of the Ophidia. They
Lasers
didn't get
off.
a clear
Thank
look
you.
at those on that fish in the image. But it's just a guess. It most closely resembled a species described from the Atlantic Ocean. And the question is that a group of similar species, or are we getting some new range extension records from these observations?
Okay.
We'll have to look at the images from that
zoom
video
out.
more closely to come up with an identification. So, Chris, I'll let you talk about the sponge now.
Sure, yeah, no, thank you very much, Bruce. And that's Bruce Mundy. He is one of the premier fish biologists that's at the Inouye Regional Center. He's part of the NOAA Fisheries Group in Honolulu on Ford Island. So thank you, Bruce. The sponge we are looking at, I'm pretty sure, is in a family that's called Eurydidae, and it's in the genus Conolasma. And again, it's a glass sponge. We're going to see, most of the sponges we see are going to be glass sponges here, which means that they have siliceous skeletal parts that give them this very glassy.
So Nav, I assume you've already been through the exercise, but you confirmed a bailout plan with the ship.
Yes, but Fernando's setup is fine. It's 150.
Okay.
Right off the slope.
All right.
It's sideways for the ship, but
Okay.
there's really no other option.
And there's a bit of a current this morning. If we did lose control of the ship, which direction
There's
would
not
we
much.
drift?
We would go astern.
Sort of
which
a
would
ratty,
be,
one of these, I think,
parallel
Calyptrophora
to
angularis
the
that we've
slope.
seen before. It doesn't look
Okay.
particularly healthy.
Zoom in a bit.
It's got a lot of brittle stars on it for such a small colony.
And hold
Maybe
there.
they're affecting it a bit.
Zoom in tighter. Hold. Any interest in getting a closer look, or is this fine for you?
This is fine, thank you, pilot.
Okay. Okay, let's zoom out.
Got a tiny little stock sponge there that's sort of in the center right. And again, the substrate's kind of a mixture of consolidated rock with some cobbles and things in between there as well, so it's not all cobbly like when we first got down.
Now this looks more massive again, and definitely coated in a thick layer. So your earlier question about how much manganese, well, this is coated with manganese very substantially. You can sort of see individual rocks are sort of hidden under some of these bumps, and so it's pretty thick, which is kind of in line with taking millions and millions of years to increase this layer thickness. Is that another?
Yeah, this is a different sponge, and maybe we could take a look at this.
Okay.
So this is it's in the same group. It's a glass sponge for sure. I think everybody now kind of can get the idea that they're going to be sort of whitish or translucent. But this is clearly a vase. One we just saw was kind of a leafy one, and some of the other ones we saw were kind of more lobate, larger white ones. This is our first vase. And we'll take a closer look and see if we can narrow down perhaps.
I know, Roland, you're going to want to look down the top, huh?
Typically, I like to see that net over the top.
Yeah, I'll see what I can do.
Because that's what they end up using that in the base.
Yeah.
Often to determine what it is.
How are we looking on the move?
Chris,
is it over?
is there a particular advantage to this type of body design?
Yeah, almost done swinging, I think.
Okay.
I'd be okay with another.
Boy, you would ask me that, wouldn't you? You know, I don't know, but there's one thing about these vase sponges. There's an older hypothesis about them, which I'll mention in just a second. So, who are you?
Zoom in tighter. Hold there.
So it's one of two things here. It's either in the genus Dictyalis or it's in the genus Regadrella. I think both of those are in the family Euplectellidae. They're sometimes very difficult to differentiate the two. You see these little spines right on the opening of the tube itself. That's called the osculum, and then these sort of projections that are covering the opening is called a sieve plate. So this is sort of a very sparse sieve plate. The other thing we kind of look at is sort of the framework itself, and you can, in the regularity of the hole. So in this particular sponge, the water's coming in the sides of the sponge, and then it's collecting in the center and going out through the top. And that's how this thing filters the water. So literally the water enters into the side. That's where the living part of the sponge is located. And yeah, there's an interesting shrimp. I'm not sure he can get out anymore. He may be trapped in there for life. But because of the shape of the sponge, some people have speculated that they can actually create a Venturi effect where the water current moving laterally will come across the top of that, kind of like blowing over a Coke bottle, and it goes over the top of that and actually pulls the water out at a faster rate, which allows it to accelerate its filtering ability and therefore eat more.
So effectively it's a large biological chimney.
Yes. So I think this is more likely to be a Dictyalis. I'm going to go ahead and mark that in as what it is. And as far as that transparent
I'm losing it.
Crustacean is, I don't know what that is for that shrimp. Beautiful imagery of it. So maybe Mary
cool
Wickstein will be able to figure that out.
Very nice shot.
Okay.
So to keep on pace, we need to be putting a 30-meter move in every maybe 11 or 12 minutes.
Okay, so we're doing fine then.
So we're currently at around 2,262 meters or so as we're progressing up this ledge.
And also just remember at the top it flattens out.
Yes, promontory,
so
I guess.
It'd be easy to move fast up there. Me too.
So we're getting sort of a smattering of animals here and there. This is kind of, I guess, a modest community at this particular point on the Seamount. And A lot of times this is pretty much the densities you see in many places, and what we're hoping to see, of course, is to encounter a higher density community a little further up. But who knows? This may be the way it is all the way up.
All right, maybe we should go ahead and put that move in.
Okay. You like the same bearing?
Yeah, it works
Yeah.
for me. Looks good somewhere.
That could have been some type of a bamboo coral.
Bridge, ROV
Do
nav.
you want to take a look at it real quick?
Sure.
Okay.
Yeah, because I think
Go
it
ahead.
is a little different.
We'd like to
passed
repeat the
one
last
up
move again.
earlier.
30 meters, bearing 340 degrees, 0.2 knots.
30 meters, bearing 340 at
Bamboos,
0.2.
They got
Good
their
copy.
name because they have these little black, what they call nodes along their branches that are spaced out different distances, and it gives it sort of a bamboo-y appearance.
And do a snap zoom roll. Hold there.
And I do think this is a bamboo.
Sorry, I'll get a closer look here. Go ahead and zoom in. Hold there.
No, I don't know. I don't think so. I think I blew that ID. I think this is another primnoid in the family Primnoidae. It's an interesting double-branched primnoid.
Nav, bridge, move initiated.
And it's got a friend, it looks like.
Yeah, and it's got a sea lily, or excuse me, a feather star that's attached to it on the top there. Yeah, it's a primnoid, all right. So I sort of blew that one. That's why it really helps sometimes to get a closer look at these things, because a lot of them can be quite similar. Diva Amon, she has identified this this feather star as being in the Komachulity group. Sako
Okay, zoom out.
Matsumoto is with us on the chat room from Japan, from Chiba Institute of Technology. Thank you, Asako, and she also has
Thank you,
identified this coral as a primnoid.
Video swapping out.
Okay, thank you, Roland.
So in the meantime, it looks like we've gotten back into a little pocket with more of these smaller, loose, broken up pieces of rock as we're sort of transiting to the next larger boulder structure. Looks like we have a few things here, Chris.
In front of us is definitely an ice-fitted. I won't blow that ID this time.
What would you like a closer look on? Got a few. We've got a fan on the right.
Yeah, let's look at this fan here on the right. Okay. That's something new. Yeah, it looks like it's got some things growing over it. So, and we've got a sea cucumber as well, and so I think this fan is going to be a primnoid, possibly in the genus Narella. And these kind of leggy fans in the back, and perhaps the double branch one that we just imaged, are also in the genus Narella. But I'm not really sure. I wonder if anyone has got another opinion.
All right, let's just zoom in here. Hold.
Holding partial.
Z-Bi is just...
We're waiting to see if Sako, she's identified it as an octocoral, whether or not she'd agree this is a primnoid. And there's a sponge, probably one of these Tretopleura sponges there in the background.
Try to face into it more.
Yeah, and so we've got a Barrett Nolan on the chat room with us. And
I'll push in.
he's asking about what's on that primnoid. And yeah, we think these either are zoanthids or anemones, Barrett. Yeah, we've got a Polythroidian, the sea cucumber that we just passed. And these, again, I think these leggy corals are... also in the Narella, and they could be something that we call Narella macrocalyx.
Yeah, pretty deep. Yeah, Chris.
Oh, there's a fan right to the right of the boulder that's new.
I see it down there.
Yeah, that would be a good one.
Just noticed a little bit of an increase in the current here.
Can you tell me what direction the current is coming from?
Well, it's coming from the west to the east, roughly.
Okay.
And we'll go zoom in on this coral down here.
Something right between your skids, too.
Yep. Sea cucumber.
Yep.
So we've also got some other people on the chat room. We've got Charlie Wall, who's at the NOAA Marine Protected Areas Center, who's participating. Hello, Charlie. And we have Deborah Glickson from Harbor Branch, who's on, and Kevin Kokot and Michael Vecchione. They're also on.
Okay,
along with Sako Matsumoto, who's been a huge help with identifying a lot of these corals this past year on these cruises.
Go in more. And hold there.
So Sako has identified this coral as a chrysogorgid.
Hold.
It's just another type of coral, first one we've seen on this site. It's an octocoral, which means it has 8 tentacles. And if you were to count all those tentacles coming off of each of the polyps, you'll see that each of them have 8. And that again narrows down what type of cnidarian it is.
Move is complete.
Okay.
Thank you, Dan.
I think it's in the genus Chrysogorgia. And
the
the little squat lobster right on it, I think, is
further west, which agrees with high pack.
That we're seeing is underside.
Roger. Coming full way.
Okay.
So, yeah, thank you very much. That was good. So we're definitely getting a variety here. You know,
are we full white?
Yes,
we're not getting high densities, but we're certainly getting diversity of animals at this location. Kevin Kokot has identified that squat lobster as being in the Galathioideae.
Why don't we swing your heading over, co-pilot, and
center it up?
Yeah.
Copy that.
And I'll center up in your camera.
So I'll give you a little update on our environmentals here. We're currently at 2,250 meters. Our water temperature is 1.9 degrees centigrade. Salinity is 34.6. And where's our oxygen? I don't see oxygen. What's that? Oh, there it is. Okay. Our dissolved oxygen is 3.45 milligrams per liter.
Fans on this rock here. You still swinging a bit, Propel?
Yep.
Yeah,
I got
so
some
Chris,
time, huh?
this is Bruce. Just for context, that temperature that you gave in centigrade is about 36 degrees Fahrenheit. So that's only about 4 degrees
more
above
creatures over
freezing.
here.
This is an extreme environment for these animals with very cold temperatures and high pressures and no sunlight.
Yeah, thanks very much, Bruce. That was Bruce Mundy at the Noye Regional Center. And yeah, it may amaze some of the viewers who are new to these particular dives how we can get so many animals living under those conditions.
I got you at 65
but
degrees.
actually, most of the ocean
67.
is pretty cold and pretty dark and very high pressure. And so it's just something that we find inhospitable. But a lot of other animals certainly seem to thrive under these conditions.
Any interest here?
It's a dead Tretopleura sponge. I think we could go ahead and move on, and I think these are the same type of primnoid corals. It's dead or dying. It's on its way out with that brown coloration.
Copy. Sonar looks happy.
Yeah, just got a little local feature here.
Well, there's a sponge down in the crack. Let's see what, see if that's anything different that we haven't seen so far today. I think it's going to be one of these glass sponges in the genus Poliopogon.
Hold there.
Yeah, it's hard to see how it's attached, but I know just from its shape that this is going to be one of these fibrous attachment sponges.
Okay, is that good enough of a view for you, Chris?
Yeah, that'll work.
All right,
Thank
let's
you.
zoom out.
That's a nice illustration of these big rocks. Even if the volcano is no longer active, the cracks show you that these pieces can sometimes fall over. It only needs a little bit more. And that big block that sponge is sitting on will be sliding downhill. It's pretty dramatic rocks here.
Yeah,
A lot
very
of
dramatic.
creatures at the top of this thing.
You want me to turn?
Yeah, are you settled out?
Almost, yeah.
Yeah, sure.
All
that's
of
fine.
these corals, it looks like, are primnoids again. There's a couple different types. There's that Calyptrophora angularis, or at least what we think is Calyptrophora angularis on the right-hand side. On the left-hand side, these kind of leggy ones, I think these are in the genus Narella. That's my guess.
Yeah, you can
That's
see
pretty
the
good
current just
scene.
blowing
Looks like little
past.
spires coming up, corals on top of them, really.
It's a nice shot. Anything you want to zoom in on?
I think we've seen all of those.
It's like a little antenna rock.
Yeah. Yeah, there's kind of one in the back there, Pilot, that maybe is a little different. That sort of very bushy one.
The fan kind
yeah,
of
the
center
little
screen.
fan thing. We may or may not have seen it, but it might be worth taking a look.
You happy, co-pilot? All settled out?
Yeah, I'm all settled out.
Okay.
We can turn to 330 and then call move, I'm thinking.
Okay.
Yeah, so Barrett Nolan had noted that there's a stock sponge in the back there, and
I'm gonna zoom in. And hold.
so this is primnoid, and
Zoom
my
in tighter.
guess is it's in the genus Narella, but
Hold there.
see if anyone else has a different opinion on what this is. It could also be in the genus Candidella, I suppose. So maybe it's safer just to stick with primnoid. It's almost certainly in that family, and Asako Matsumoto also agrees that that's the correct family.
Okay. Seamount.
So we may or may not have a number of aquariums watching today. The Maui Ocean Center on the island of Maui in Hawaii routinely broadcasts the footage, the live feed, in what they call the
So you want to go
deep sea
330?
cave.
Yep. Okay.
And so if
Push
they're
over.
tuning in this morning, welcome to the visitors there at the Maui Ocean Center. Hope you're enjoying this live exploration that's occurring right now off of the NOAA ship Okeanos Explorer.
What's that?
We're making you nervous or moving
We also have the Waikiki Aquarium that also broadcasts these the feed here as well. So welcome to the visitors to the Waikiki Aquarium who are watching this, and sometimes as well the Aquarium of the Pacific in Long Beach. So more and more of these aquariums are starting to pick up, and sometimes as well the Aquarium of the Pacific in Long Beach. So more and more of these aquariums are starting to pick up on the idea of telepresence, these live broadcasts.
If you're ready, pilot.
Let's just wait a minute. Okay. Just come up a bit more and make sure there's nothing they want to look at.
Sounds good.
Co-pilot, I just can't help admiring that wire drum over there. It's beautiful.
That is correct. Might be a
Statement
little
of fact.
We had a couple of Asako and Barrett noted a sponge that we passed over, and I think that sponge was in the genus Phariid, in the family Phariidae. Oh, look at this one. Now we've got a nice looking sponge here. Might be worth a look. I think it's the first time we've seen this one. Yeah, very angular rocks here. Again, they don't look like pillows to me, Jasper. What do you think as they're getting ready to zoom in here on the sponge?
Yeah, this looks more broken up. These are definitely fragments. This is not complete pillow structures. This is more broken up material. So the lava usually, after it cools, it can crack, whether it's pillows or flows, it can crack and leave more angular pieces.
You can zoom in.
I'll let you take a closer look at this guy.
Yeah, so we've seen this before, and I'm not absolutely positive. It could be something in the family Euplectellidae, and in sort of what's called a subfamily Corbitellinae, which are the vase-shaped Euplectellids. And there's something that this is similar to that we call Corbitellinae new genus, because we don't know what it is, even though some specimens have been collected off of Hawaii.
I have a lot more zoom if needed.
Go ahead.
So I'm going to go with that.
Zoom in. Hold there.
Things you look at here are just the arrangement. See all of the projections coming off of the side of the sponge. That's typical of this Corbitellinae new genus. It's informative. Maybe at the base where it's attached, if we could take a little quick look at that, that brown.
Go ahead. Hold there.
Yeah, so this is a solid sort of glued type of attachment that we call basophytus attachment. So it's not fibrous. That's what I was looking for. So I'm fairly certain the family is correct on this Euplectellidae.
Okay, let's come out.
But then after that, boy, I don't know.
Full wide.
Thank you.
Yeah, thank you very much for the.
All right, I can call that move in.
Okay. Bridge, ROV nav.
gives you a nice view of how steep the terrain is that we're on.
Bridge, ROV nav.
Yeah, from our original dive plan, it looks like the first
Go ahead.
little bit
We'd
over
like
the
another
first
ship move,
half
please.
is
30
sort of
meters,
steeper
bearing
section, and after that it's
330
gonna
degrees,
level
0
off a
decimal
little
2
bit.
knots.
This is definitely still part of a
Ship
steep
move
set.
30 meters, bearing 330 degrees, speed 0.2.
Good copy.
There's another Sacco calyx and another, it's like either a Regadrella or a Dictyalis. Particular Sacco calyx has got a large anemone on it. I think maybe we could take a look at that. And I think this could be a different anemone that we haven't seen before on this particular dive. So there's a couple different types of anemones that are generally referred to as Venus flytrap anemones, just because they kind of work somewhat similar to the Venus flytrap plant. They tend to be folded over and form this sort of trap, and animals that are unfortunate enough to swim in between the two lobes, they wind up closing up and getting trapped and consumed.
Lighting's not going to be great, but we'll do it.
So this particular Venus flytrap anemone, I think it's in the genus Actinoscyphia. And I'm going to go ahead and again tentatively identify it as that.
Hold there.
So really, really beautiful anemones. They're quite large, but they're just gorgeous animals down here. And these particular Actinoscyphia are off and on other types of animals like sponges and corals. They're a type of anemone that presumably doesn't do that well on the ground. It needs to get up into the water column to successfully make a living, if you will.
Yeah, so he's just using this guy as a ladder to hang off of.
That's correct. So he's not hurting the sponge. This is sort of a
Would
very
it be all right to go in a little tighter?
commensal relationship.
Sure,
I'm not sure the sponge gets any benefit from the anemone, but
Can I
clearly
go
the
down a little
anemone is
harder?
benefiting from the sponge.
Try not to move. Go ahead.
You know, Chris, this is Bruce.
Okay, I'll
I
hold
wonder
there.
if
It's
the
not quite full.
anemone, since most anemones have nematocysts or stinging cells, if the anemone would help fend off any sea stars, because some of the sea stars are predators on sponges, and if a sea star tried to climb up that stalk to eat the sponge, it might encounter the anemone. So there could be some benefit, but that's just speculation.
Well, it's pretty good speculation, Bruce. I hadn't thought of that. So, well, thanks very much. Yeah, absolutely. You definitely get sea stars that are predatory down here on sponges and corals, and you may be exactly right that the sponge is getting benefit. The sea star, really, to affect the sponge, has got to get up to that lobe, and it's got to go through that anemone before it can get up to the actual living part of the sponge.
Quick establishing.
You got another. Oh yeah, we got some stuff in the distance there. It looks like a larger sponge of
Okay,
coral.
we'll move on. I'll come back over.
And Asako had noticed the hydroid on the sponge stalk.
Yeah, we
I
can get
missed
that. I think maybe at the end of your leash, but
what is this creature?
Just let me
It's
know
a
if
really
you need me to
whopping
do anything,
big
co-pilot.
anemone, but I'm not
Copy.
sure.
Looks good to me right now. What is that?
I'll turn a little bit to starboard to keep you in view here.
Okay.
All right, who's going to
Looked
get
like
this
that
first?
last move ever
We're not
happened.
hearing a
Talk
whole
to
lot
the
of
bridge.
IDs. Chat room is silent.
Any volunteers?
Yeah, we're not.
Yeah, I don't have a whole lot of tether here. You want to snap zoom?
I want to talk to the
Zoom.
bridge.
Sure.
It's kind of an enemy.
Bridge, ROV nav.
So it's
Go
an
ahead.
enemy.
Did we ever get that last move started? It looks like we're stationary.
It's a big one. Do we have the lasers on by any chance?
Yeah, we can get lasers on.
Apologies, as training
I'll
up
fill the
here.
frame.
I'll,
Lasers on?
Wow,
let it
so
now.
those
That is great, thank
lasers
you.
are 10 centimeters across, if I'm not mistaken.
They are 10 centimeters.
Yeah, so that is one big anemone. That looks like it's 20 to 30 centimeters across on the disc.
Bridge move initiated.
Next bridge.
Okay, let's zoom out.
And as far as going past an enemy, nope. We're going to leave it as an enemy.
Got enough leash.
That's going to be close.
Yeah, we're starting
and
to
I
move
haven't
now.
either.
That's
And that was
up there.
330 or 340?
Yes, 330.
Okay.
Yeah, that's the comment in the chat room right now, is
think that's
that
about
no
all
one's
we got.
really seen this thing
Yeah,
before.
okay.
Yeah.
It's-
Well, we're going to have to wait to get a little more tether before we get a closer look, but
Okay. Well, this is unusual, so yeah, it's worth getting a little another look at this thing.
absolutely. Well, I'm just going to saddle to the left here. We'll see what else we see until we catch up.
Okay. Another one of these glass sponges. Could be in that Corbitellinae new genus, I'm not really certain. Or it could be something different. It's actually starting to look a bit different.
Co-pilot, you should be starting to get closer now.
Copy.
Maybe
Okay,
give
you want
it
to zoom
another
in real quick?
minute or two.
We'll get this,
Sure.
and I'll move back over. Hold there. That's similar to the one we saw before.
Yeah, it is similar. I agree. I just couldn't quite see the lobes until you zoomed in, but yeah, I think it's potentially a Corbitellinae, but there's another possibility. It could be a hard sponge too in the genus Perifragella.
Okay. So now I'm going to move back over. I'm excited to see this thing.
Video, Copilot. How's the lighting? Is it too hot in the middle, or is it okay? Okay, thanks. Yeah.
It's kind of a nice side view there. You can see a little bit of the stock. Hard to say whether the tentacles are retracted or whether they're just these big, massive tentacles coming off.
Could
of
we
this.
turn off the
huge.
lasers?
Yeah, we can go ahead and turn them off.
So yeah, that's about 30
That
centimeters
laser's off. It's
tall.
the biggest
too.
one I've ever seen.
So, yeah, that could be a record for me too, pilot. That's a big one.
Okay to zoom.
Hold on 1 second. Okay, go ahead. Hold there. And it's a little bit hot. Yeah, there we go.
So we're looking at the chat room right now, trying to see if anybody out there really has an idea of what we're looking at here, aside from an enemy. We've all concluded that we've gotten that far.
The geologist might have even guessed that one.
I know. It's not manganese crusted though, Jasper.
That's right. It must be younger than a million years.
Right.
All right.
Yeah,
Go
that's
tighter.
a really gorgeous view of this. I mean, it's there's certain features that look similar to the
It's not
family
quite full. I'm just gonna
Actinostolidae,
hold here for a second.
Okay.
but I just don't understand what's going on with these tentacles at all.
Get my toe back here. Oh,
Yeah,
sorry,
that's
Tara.
just a
No
marvelous
worries,
view
that
at
was
that.
lovely.
So
I can
anybody
do it
knows
better now.
out there, you know, once we
Just
get
trying
these
to get this
pictures
auto-heading.
distributed.
There we go. Feeding him.
So the thing
Lunch.
that's a little confusing, it has a very textured stalk here that's almost almost Hormathiid-like, but I don't
Live
know.
on the other
I'm
side
not
of him here.
sure it's a, I don't think it's a Hormathiid. So
Zoom out partial. Hold there.
So, Chris, it's unusual to see the ends come into these little rounded tips with a little brown point in the middle there.
yeah, I
taking a
just
while to
haven't
recover
seen
from
that
that.
before, and I don't think anyone has. Just again looking at the chat here, Diva's never seen it before. Osaka's never seen it before. Since Charles Wall was commenting that, yeah, it's an anemone, but it's about as far as he was able to go on it. And same with Bruce, so yeah, it's kind of stumping us all
You're
here.
in tighter. Hold.
If we could go closer on the edge of the tentacles, I can get a closer shot with the dark background.
Yeah, I see.
Bright
So it
blue.
looks like the little division,
Go
the
ahead.
tentacle there is splitting or something.
Yeah, it looks like it's splitting. It's like a fork.
Yeah, so what's up with that? I mean, I don't.
It brought its fork for lunch.
This is really quite unusual. Yeah, and Barrett Nolan had sort of commented that perhaps they're retracted, I guess. Retracted to a partial extent. It didn't really do anything more when the thrusters actually, created a little bit of current there. So
Okay. Video's clear
yeah,
whenever.
very, very unusual, very cool.
Hold it here.
Roger.
Right there's good. Any other part of the organism you want to look at?
No, I think I think that'll do it, and
Okay.
it's just a matter of us getting some still captures from this and getting them to some
Okay,
people
let me
that
zoom
might
out
have
and
an
frame it up.
idea.
Turn.
Yep.
Hold there. Nice.
Nice shot.
Yeah, thank you very much for that.
Yeah, so he was about 30
Well-
centimeters across or so.
Amazing.
So it could be something in the family Exoselactinidae.
All right.
Just
Coming
looking
away.
at some
Coming away.
pictures here, trying to find anything
Nice.
that's somewhat similar to this anemone.
You're off to my starboard.
Yeah.
Ship is stationary.
Ship is stationary. Okay.
I got a little bit of swing left, probably.
I'm just going to move over.
So that picture you're
And
looking
then
at
leave,
is
you and I are
different in
gonna
color.
switch.
Is that in any way
Does that sound good to
helpful
you?
in identifying
Sounds good.
any of these guys? Or is that not at all distinguishing?
When maybe in a species or possibly genus level, but when we can only go to family, I don't know, the family can have all different types of colors there. So
It's definitely not bad, but there's a little bit of current today.
Copy.
Kind of coming from, just port to starboard. If you're looking
Ship
up
was
slope.
330, I think. So
Yes, it was.
I'll turn to that real quick. I turned a little bit to starboard to keep you in view.
Sure.
330 looks like it continues to be upslope.
Copy.
Okay.
Pull up a little bit.
So in the meantime, it looks like we've leveled off a little bit again in topography, and we're on some smaller rocks again. We're looking around for some other features here.
Okay, so So you're looking good. I'll push out a bit.
Okay.
What is that? Sponge stock or something?
Yeah, the chat window, they're saying these are old stocks.
Yeah, well, let's just do this quick. Just get pushed around. But I'll come up. Man, let's push it hard.
We are just falling behind that .08 average we need to make.
All right. Well, not too worried about it because it's be pretty flat up
Right.
pretty soon. Thanks for keeping an eye on it. All right.
It's going to blow you up to the starboard as soon as you pick it up.
All right. So you can go ahead and come up and talk to Watch Lead and see what they want to do.
Just get a feel for the current real quick.
Check, check, check. Mic, check. There we go.
You're good.
So the laser's light on the panel doesn't go out.
Yeah, John.
Which is actually kind of nice because then you know exactly which one it is.
Yeah, I was thinking of marking it with some tape, maybe.
So the chat room is trying to still look at some of the creatures on this central rock. We've had a few folks shuffle through here. It's right around lunchtime here, so we're doing some quick shift changes. There's some ideas of what might be what here. Though I'm the geologist, so it's hard for me to
So
see
you got
who's talking about
50%
what.
thrust just kind of holding steady.
Yeah, copy that.
So you're going to have to be mindful of that.
Okay, I'll come in closer here.
Just notice we got black bars on each side of the
Copy that.
image.
We do.
Never noticed that.
Copilot, you want to take my
So in the chat room, they're asking whether
take your camera.
whether
Yeah,
these things
I
are
got it.
attached to each other somehow. Maybe we can take a quick look.
And video, feel free to come in. It's
Maybe
Roger.
from bottom to
top,
top.
bottom, top, whatever you guys want.
Holding partial.
Just let me know what you
Just
need.
for a second.
Start with the bottom here.
Okay.
Okay. I'll just.
If you sit down, I can
take
tilt
a look
up
above the sea star as well.
if you want.
Might be too low, but I'll might be out of the light pool. I'll just get closer.
So we're trying to make our way up this creature.
Just watch this rock. It's coming in between your toes.
Yeah. Copy.
Shoreside scientist, what's the... I
Pilot, if
can't
you could
tell
back
what
up just a
is
touch.
that a sponge that's
We're losing
just
the light.
above the Brisingid that
There.
has the Zoranthids on it?
Nice.
I'm having trouble
I'll
telling
come
what that is. Does
partial
anyone on
when
shore
you're
have an idea?
comfortable. Or a little tighter zoom when you're comfortable.
Yeah. Just the current's blowing me around a little bit.
Yep, absolutely.
I'm worried that if we zoom in, then.
Roger, I'll hold here then.
Thank you.
Craft shoulders sagging a bit.
We're not getting any
Nobody else is answering,
suggestions from shore either right now.
This is Bruce, and
Hey, Bruce, you're a little quiet and broken. I don't know if it's our connection or if you're just a little bit far away from your microphone.
yeah, I see nobody else is answering, but I'm the fish biologist, so I don't know. I see the sea star, but I haven't seen what looked like zoanthids yet. I'm at a little disadvantage here, though, because I'm seeing the real-time broadcast on a
Just
small
gonna
screen. And
come out just a touch so I can keep it in the frame.
I'm using the internet one, I think it is, for my big screen view, which has a lag of about a minute behind what we're seeing on the small screen. You know, I don't know if Viva's still there. She might have more of an idea of what we're looking at.
I'll come up to the.
Diva, for the Pharea sponge, is that the one at the bottom, or are you
Northern
saying that the one above the Brisingid, between the Ophiuroid and the Brisingid, are is the Pharea?
Is the lighting poor just because it's so close?
So.
Yeah.
Hey guys, it's Viva. So I think what we're looking at is a dead sponge structure, something like the Tritopluras maybe that Chris is so familiar with. And then we've got one large Friella Brisingid sea star, I think.
Watch your.
And then
watch
also the one above it looks like an Ophiuroid, maybe from the family Okeanos. And then you can see, I think there's still a bit of live sponge tissue just in the middle of the screen right now. And it looks like it has some anemones growing on it, maybe. They're some type of little polyp type animals. So I'm going to guess anemones.
Thanks,
I
Diva.
think that's all the things I'm seeing
Because
right now.
it was the little bit of live sponge tissue that was confusing me, that I was having trouble figuring out what we were looking at.
Okay, watch. Is there any specific item on here you want me to zoom in on, or would like keep moving here? Copy that. Come out, video. Thank you. I'll take my rotator back.
Okay. So I'm 322. We want to go 330, correct?
Yeah.
What do you think, Nav? You want to shift more westerly?
320 might be okay.
320. Looks fine to me.
Looks Good based on the sonar. No, let's do 3-3 again.
Okay. Why don't you push out a bit, get the move underway. I'm at 330.
Can I get lasers?
Lasers off.
Oh, they're on?
So we're moving
yeah,
moving on along the little area with the looser rocks here. That looks like something that has died a while back in the middle there.
Yeah, let's go ahead and call it in, Dave.
Okay.
We're ready.
Bridge, ROV nav.
Go ahead.
We'd like to repeat the last move. 30 meters, bearing 330 degrees, speed 0.2 knots.
There's
30 meters bearing 330 at 0.2.
guy right there,
Copy.
right in the middle for us at this point.
Move initiated.
Maybe we can take a quick, closer look.
Okay, videos. Sounds like they just want to take a quick look at this guy.
Roger. Standing by to zoom.
Yeah, feel free to zoom.
Okay, I just came back from a good lunch here, and we're looking at a sponge, which is terrific. So this sponge is probably in the genus Bolosoma, and it's a euplectellid. It's a soft sponge, but it's a stalked soft sponge here. And some of the cues we have, again, is the way it attaches to the rocks, where the laser is, the actual shape, how the stalk comes into the upper part of the body. These are all things that we look at when we're trying to identify these sponges. How thick the walls are, so we can, this is a really great image, so I'm fairly comfortable with going with that genus on this one.
Would you like more imaging on this pilot, or are you good?
Watch lead pilot. Yeah, watch these. Did you get enough documentation of this guy?
Pilot, were you talking to me?
Yeah.
I couldn't hear you, sorry.
Did you get enough of this? Or would you like me to zoom on any specific parts of it? Or?
Yeah, we have enough of this, thank you.
Okay, you can pull out video.
You know, it helps to actually put my my ear part of the microphone here actually on my ear. I just discovered you can hear better that way. It's really funny how that works. It's kind of funny how you don't work as well the older you get. I was wondering why I couldn't hear anything, because it was sitting on top of my ear. Anyway.
So are we underway now?
There's a long-legged shrimp in the Meadocarcinus, I think.
20 meters left in
Yep,
this move.
looks like it, because the way he's kind of propped up above the substrate.
Push out, pilot.
Tell from here, but we'll...
A little bit.
Yeah. Do you need an ID on this shrimp, or we kind of need to push out?
I think we can keep going. This is common everywhere, it seems. And looks like we've got some more primnoid corals and something else there, I guess.
Which one are you interested in, watch lead?
Maybe this one right directly, this unbranched coral here. Maybe we could take a peek at this. I think it's could be a coral in the family Primnoidae, but sometimes they're really confusing with the bamboo corals.
Okay, you can frame it up.
This could definitely be a bamboo, and I think behind it is a stalk crinoid that might be worth taking a peek at as well. So this looks to me like it is a
Next note.
bamboo
Holding partial.
coral. I'm going to see if anyone...
I'm just going to put my toe down and then I can get a
Roger.
Yeah, boy, I don't know if it's a bamboo coral at all. Maybe not.
Just do it floating, actually.
It's got 3 polyps coming off the stem, so maybe Diva and Asako, maybe this is a Candidella, perhaps, in the Primnoidy? See if they have an opinion on this.
Video, you can come in just a little bit.
Pardon, pilot?
You can come in.
Roger.
Zoom in just a little bit more. I'm going to try to do it floating here.
Okay, hold there. It's not quite full zoom.
Okay.
They might be able to see the polyps.
Well, I don't think it's a bamboo. I think it's definitely a Primnoid.
Coming wider.
Yeah, come wide.
And the question is what genus it is.
Settle down here.
Full wet.
Port swinger should be
So Barrett Nolan has noted that there's a stalk crinoid behind this coral.
Okay, video, come in.
And
Co-pilot, take a rotator.
the
Do
crinoids
whatever
are
video wants with it. I'll try to keep my toe in.
were used to all be considered, I guess, sea lilies, or at least I used to consider them all sea
I'll start on
lilies.
the left side and then I'll work my
Holding
way
partial.
to the right.
Take a look at that after we get a good look at this coral, which is a little confusing. Asako thinks it could be a Primnoid.
Okay, I'll take a bit more zoom.
Yeah,
That's
now's your time to zoom here.
Roger.
I would sort of...
Still, I'm going to be able to get it in this position.
yeah, this is a really nice, beautiful shot of this Primnoid. So I think this could be something similar to Candidella gigantea, just by the size of the actual polyps and the fact that there's 3 of them coming off at each whorl, and that it's unbranched. So that was a fabulous view of that. Probably the best view of one of those
Okay, I'll get a quick
types of
stab.
corals
We're gonna wanna
that you've ever
keep
gotten.
going.
It's
Copy that.
gorgeous.
We'll pick up here.
And I wonder if we could get a snap
you
zoom
clear?
on that little
Roger.
crinoid to the right.
Thank you, video.
Yep.
All right, moving on.
Might want to tell watch lead that we need to push out.
So I was saying about the crinoids, the ones that have stalks are called sea lilies,
Okay.
and then the ones that are unstalked are called
The
feather
ship is
stars.
stationary,
But they're
Yeah.
all
probably can swing a bit.
part of the same group.
Watch lead pilot.
Roger, pilot, go ahead.
Is there any specific item you want me to zoom in on next? Else I kind of need to keep moving here too.
How about the sponge that's right up ahead on the stalk? I don't think we've documented this type of sponge yet.
Absolutely.
Yeah, thank you.
Yeah.
So this stalk sponge, I think, is in the genus Calophacus,
So I keep my
and
toe on
it's
this rock.
not a euplectellid. It's in another family. And again, things that you look at is are how it attaches to the rock, how the stalk attaches to the body of the sponge up top, and in the Calophacus, the stalk actually comes up and then goes into the back side of the sponge, as
Okay,
you
go
can
by,
see
you can take
there.
Rotator again.
So
And video, feel free to zoom.
those are all clues
And then start at
as
the bottom, work
to
the way up maybe.
what type
Roger,
of sponge
sounds
it
good.
is.
Tilting
You
down.
also look at the thickness of the stalks.
Holding.
Oh, sorry about that.
Yeah, so you can see that it's got sort of a glued-on plate here. That's how it's attaching to the substrate. So that also is very informative. Sponges attach to the substrate differently. This guy basically creates a plate of silica that it sticks to the rock.
Okay, ready to tilt
Other
up?
sponges actually have these fibers that are coming down that are called basalia. They're a specialized kind of skeletal component or spicule that has little hooks on it, and they sort of grapple the substrate, and that's how they hold on. And there's other types of sponges that actually live in the sediment, and they can have roots, or they can have also these fibrous attachments. And here's a beautiful pan right up the stalk and to the actual body of the sponge. So they consider this type of sponge to sort of be inverted, so the actual right side of the sponge is considered what's called the atrial surface of it, and in vase sponges, that's the inside part of the sponge, so it's as though in this particular species, the inside has been flipped over, almost like a glove that you take off and you sort of flip the inside over. And the actual exterior surface of it is where the stalk's coming in on the backside. So again, these are things that sponge
maybe I'll go in a bit
look at
tighter,
to try
and
to
if you could just tilt,
identify.
I'm going to try to rotate
these
right a little
different
bit.
species. You can kind of see those little
Slip off the rocks.
rods and things in the texture right now. Those are the spicules or skeletal parts of the sponge, and that's what most of the glass sponge taxonomy is based on right now, although they're moving toward genetics. But the way you would analyze this type of sponge is you would take a small piece and you would put it in bleach or some type of an acid and dissolve away the tissue, and that'll loosen up all of these skeletal parts, and then you look at these spicules or skeletal parts, and they can be very distinctive between species and families, and you can determine what the sponge is
frame with the organism right there
under the microscope.
Nice.
Okay.
All right.
Watch lead, pilot.
Back to you.
Roger, pilot.
Yeah, did you get enough imagery of this?
Yeah, that's great. Thank you.
Let's pick up and get you ahead.
Sounds good. We'll keep moving.
So Jasper's back from eating his lunch, and Jasper, no, we didn't see any rocks while you were eating lunch.
just water and animals, huh?
Yeah, that's a dead sponge stalk.
Oh.
Right there. Oftentimes the dead ones, they kind of lose their tops. I'm not really sure why. When the top falls off, oftentimes the stalk itself remains.
Okay, we'll keep going on and up. See what we see.
Little update on our environmentals here. We're at 2,196 meters, and we are at a temperature very close to 2 degrees Celsius, which Bruce Mundy had pointed out was close to about 36 degrees Fahrenheit, so it's pretty darn cold down here. Salinity is about 34.6, 35 parts per thousand. And our dissolved oxygen is 3.37. Now that number may not mean anything to you, but basically at the surface, our oxygen levels are around 6 to 7 milligrams per liter. Down here, it's about this particular depth and location. It's a 3.4, so about half of the oxygen at the surface. And here's a really beautiful anemone. This is different than the one that we saw earlier. Oh, is that right? Oh, okay. Yeah, I stand corrected.
Watch lead pilot.
Roger, pilot.
Is this something you want to get a good zoom on, or just a identification?
We could do a snap zoom on it.
Okay.
Sure.
All right, video 50 come in. Is that do a quick one here?
Okay, I'll just hold here for a couple seconds.
Sounds good.
Yeah. So again, this is a really large anemone, but it appears to be different, unless if the other one had these tentacles contracted, then perhaps that's what explain why it looks so club-like. Yeah.
Okay, we're gonna keep moving here. See if they-
Different tactic to approach its prey. It just clubs them instead of using the.
Yeah. Shrimp comes by and it beats him over the head. Isn't that sort of a primitive way of getting your food, sort of clubbing?
Yeah.
Yeah.
There's something over there.
Another one of these vase sponges.
All ready for a move there?
Yep.
Pilot.
Yep, let's call it in.
Looks like it's a Corbitellini, what we're calling a Corbitellini.
330 still good.
Great.
I'll get just a little closer, and then I'll let you zoom in video.
Bridge, RV nav.
Okay, Copilot, you can take my rotator and
Got
video.
it.
You can zoom. And just, I'll hold it right here.
Tilting up.
So
Holding there.
Bridge, RV nav.
I'll just fill the frame first.
Sounds good.
Is there any particular part of the sponge that
Watch lead pilot.
is a priority?
Roger, pilot, go ahead.
Is there anything particular in this sponge you want me to zoom in on, or?
No, we've seen a couple of these, so this is fine.
Sounds good.
Main thing I wanted to point out to the audience here is that we believe that the water's coming in the side of this sponge as well, and then it collects in the center of it and then goes out the top. So these type of vase-like structures are fairly common in these deep water sponges. Whether or not they have these bumps all over them or not, the overall shape is apparently quite suitable for what they do, which is filtering the water.
Bridge, ROV nav.
Video's clear.
Go ahead.
Thank you, video.
Hi, can we get a ship move, please? 30 meters, bearing 330 degrees, 0.2 knots.
Okay, good copy. Stand by.
Thank you, Bridge.
Take my rotator back.
So again, those of you who are new to these dives, well, welcome. This is the NOAA ship Okeanos Explorer. We're on a seamount called Alba Seamount that's located about halfway between Guam and Wake Island, so we are really out there. Unbelievably, the water is absolute glass today.
Dead glass.
So you just don't often see the ocean looking the way it does. So we had really favorable conditions in our transit from Guam to get to this location. So we're very, very fortunate. We are currently at a depth of over 2,000 meters. So that's over 6,000 feet down, 6,000 to 7,000 feet down. And we're on an area that could potentially be mined for manganese crust in the future. And so what we're trying to do is get an idea of what types of animals and what type of communities exist on manganese crust habitats. So this is something that's been a priority of the 3-year effort by the Okeanos Explorer here in the Pacific last year, this year, and it'll continue to be a priority next year. And that is to try to document these communities because deep sea mining is something that is most likely going to occur in the future, and it's good that we get a head start on understanding the communities and what could potentially be impacted by mining activities. So we're also providing this data to the International Seabed Authority and to their leaseholders for the lease areas that we're surveying, such as this one today.
And
There's a couple of things down here we can take a look at.
And
Yes, we're in motion, so you
this
should
is potentially the
start
first
swinging
time that anybody's gotten a look at the Seamount. So we're certainly going to provide the data and the information to everybody who needs it and would like it. Got another Narella sponge here on the left, and not really sure what's in the center there, coming off of the side of that rock. But this Narella we've already imaged a few times before. I wonder.
Take a quick snap zoom video. Don't have a whole lot of image here.
Could just confirm what it is. Yeah, so this is again in that family Primnoideae. I think it's an Narella, and then going along that center branch there, you can see what appear to be either anemones or zoanthids. I think they're potentially anemones.
I have a bit more zoom. But
I can't give you more zoom. I'm kind of floating here. Yeah, try. Nice.
So these anemones do seem to disturb a little bit the growth of the coral, but not severely. As you can see, there aren't nearly as many polyps coming off of the branches where the anemones are attached. Asako Matsumoto has
video's
confirmed
clear
that this is a primnoid
Yeah, you can pull wide.
coral.
I think there's something else they wanted to look at.
Roger. Full it.
And there's sort of mostly dead one of those vase sponges that we just imaged. Little Caulophacus sponge there, that stalk sponge that we're on the boulder that we're just going over.
Try to zoom in on this little guy. I'll float right above him here. A little bit of current. I got it right here real quick.
I have a bit more.
So this is a sponge that we call Caulophacus. And again, these are this whole group of sponges are very common inhabitants down here. They really like this cold, dark, high pressure type of environment. They seem to be very successful
Video
down
switching
here
on.
as well, given the fact that there's so far we've seen these things spread throughout the Pacific.
Oh yeah.
Chris, do we know how far north or south these guys extend?
Of course,
No,
we
but
missed you,
it's
and we
something
already
that we
have
might
an
get
issue
some
for you.
information
We have
on
some black
next
bars
year
on our
because
left main
we're
screen.
planning some expeditions all the way
into that
down
if you have a
to
minute.
American
Thanks.
Samoa and to Howland and Baker, which is the most southerly extent of the 3-year campaign here in the Pacific.
Okay,
And then we're
we'll
also going to
do
get
it
up
some other
into
time then,
the
I guess.
musician
Thanks.
seamount, so that'd be sort of the furthest east.
Yeah, moving
That
toward
will
you.
be-
So
10-4, moving out.
Yeah, it'll be a very, very interesting set of data after the 3 years are completed, where we're covered all the way from Hawaii to the Marianas and all the way down to American Samoa up to the very end of the northwestern Hawaiian Islands past Midway. So there's a dead Bolosoma, very dead. He's,
You can tell by the way it is.
I think the reason they turn brown like that is they don't have any way to clear sediment that gets trapped inside of their skeletons, and so that's what you're seeing a lot of.
So maybe the whole thing just gets top heavy and the top falls off once the top is too heavy?
Yeah, that's a possibility, sure.
Push right over.
Starts accumulating debris.
Cool. All right. Ooh, hey, big fish.
Oh, look at this guy. Aha, here's your cue, Bruce. Bruce Mundy is
Siphonophore or something above it.
our resident fish expert.
Try.
He's calling in from the Inouye Regional Center, NOAA Fisheries.
Partial.
Is he coming closer?
Okay, this is one of the most common fish that we see on these deep sea surveys. This is a cutthroat eel in the family Synaphobranchidae, and this is in the genus Synaphobranchus. They're heavy-bodied eels. They're one of the larger predatory animals down in these habitats, these deep-water habitats. The species are somewhat difficult to identify. I can't get a good look now, but the position of the dorsal fin, the fin on the back, and where that starts on the back is often very helpful for identifying these. It looks like it could be one that's got a dorsal fin that starts very far back on the body, which makes the dorsal fin rather short. The species name is Synaphobranchus brevidorsalis, which means short dorsal fin. They're called cutthroat eels because the gill openings are on the bottom of the throat,
it's full wide
side of the throat, and very close to each other, which makes it look like the throat has been cut. But that's the natural state of the gill openings in these.
Came right at me.
They've got not fang-like teeth, but large teeth, and are predators. They're also scavengers, and they'll feed on large animals that die and feed on the carcasses that drift down to the bottom of the ocean. But they would also feed on shrimp and maybe even small fish that they could catch. So a cutthroat eel, Synaphobranchus, probably Synaphobranchus brevidorsalis. I'm at a little bit of a disadvantage because I get the real-time feed on a small screen here in our command center.
Keep
and
the
then,
revs
the
low.
larger view comes across on the internet feed after a minute or two delay. So I
Try to work out some of that air.
Good-sized view of that eel yet. But Chris, if you have different thoughts about the species, I'd be welcome to hear them.
No, I totally agree with you, Bruce. Yeah, you can clearly see that the dorsal fin was far back. The origin of the dorsal was far back on the body, so it certainly looked like Synaphobranchus brevidorsalis to me as well. So a real common observation that we've been making for the past couple years here is that whenever you get this sort of bouldery, cobbly type of substrate, you don't typically get that many animals, and then when you get into more consolidated substrate, the number of animals pick up, and presumably a lot of the animals prefer a much firmer type of substrate, particularly attached filter feeders. This is a Calophacus sponge on the bottom by the lasers, and we have another one of these unbranched, presumably primnoids.
You can come in just a little bit.
Above that.
Just get an ID.
It's a partial.
Could be a slightly different variety of the Calophacus. A little bit different type of head on it, as well as the actual stalk.
Watch lead pilot.
Roger, pilot.
Do you need any more of this, or are you okay with this?
I'm okay with this.
Okay.
This is good.
Thank you.
So it's got a lot of things that are growing on it.
nuclear.
Not all of those little spiky things are part of the sponge
Thank you,
itself. You can get hydrozoans growing on these things, and feather stars. And again, the point is that these larger animals can often serve as sort of a base of operations for smaller animals.
So we're still clearly on this terrain with the more blocky, rubbly sort of surface filled with sediment in between the rocks. This is sort of the thing we started with. We've seen a few larger formations. and now we're on this material again. Looks mostly broken up parts. And there's something coming back into the picture in the top there.
Yeah, maybe we could take a look at the sponge. I think it's different. You know, superficially it looks like the same thing, but I think it is a different sponge.
Yeah, we'll get in here a bit closer. We'll take a look at it.
Yep, I'm even more certain now as we're coming up on it. I'm trying to remember whether we collected one of these already, because they are different. We might have gotten one of these in the Marianas. Now I'm not so sure. What is this guy?
Okay, video can come in.
Partial.
Actually, yeah, Copilot, can you take my rotator?
Sure.
And then, yeah, maybe start at the bottom, work your way up.
And look at how thin and uniform the thickness of the
Might be
stock
on the
is
back
on
side
this
of the rock
particular
there.
one.
Yeah. Yeah, I might as well go to the top now.
Okay, coming up.
Excellent.
Looks like a straw.
Yeah,
Very thin.
it just doesn't increase the diameter as it gets taller and taller.
Minimum control.
Yeah,
I mean,
it's pretty far out there.
I guess it would decrease in diameter the taller it
Makes
got,
it
I
look long when
suppose.
you zoomed in like that. Excuse me.
Got a little wider again there.
Yeah, so you are a very strange sponge. I don't know. Let's take a little poll in the chat room. What do people
Looking out
think
for
about
full,
possibility
head.
of making our first biological collection here? I don't recognize what the sponge
Ship
is,
is
and that
stationary,
osculum
so
on
that
top, that opening was
no
sort of a
issues there.
surprise
Sorry.
to me.
There we go.
So that's going to rule out the Caulophacus, and it's also going to rule out, I mean, it could be a type of Bolosoma.
Have we lasered this yet?
It's a very
Lasers are
different
on.
one.
Yeah, lasers are going to fall behind it though, so maybe if I zoom out, we could tilt down and get the lasers up by the base.
Yeah, I think this might be worth collecting, pilot. I think this could be something new.
Copy that. I'll talk to my co-pilot real quick.
Yeah, thank you.
Going for full specimen?
Well, you do this one. Okay. It's pretty big.
Little tilt down.
Bruce is in favor.
Yeah. Okay. So we got, you know, one of the things we're doing on these dives is we're trying to keep our collections down to 2 rocks and 2 biologics. So we're really, really picky on our biological collections and even the rocks, because we want to look at the geologic history of these seamounts through the rocks. And they've got to have some basalt or something like that in the rocks in order to be able to date the rocks. And same thing for the biologics. We're looking for animals that are unique, that could be potentially be new records or new species for this region. So that's kind of it. We're going to be very, very fussy and hopefully get some really nice, interesting animals. You know, this one is one, and then hopefully we'll see something else a little further up.
Both
Yeah,
bio
and then
boxes
in
are
being
open.
fussy, you have the advantage of actually being able to see what
whatever
what
one
it
you
is you're
want.
collecting. We have the layer of manganese on top of everything, so that makes everything a bit of a guessing game.
I want to confirm.
Yeah,
that
so
they're
one of the
aligned.
things you look for is whether it has sort of angles and flat
Angles
faces
Yeah.
and things like that,
That's
right?
good to
That might
confirm.
Yes,
suggest
the only
it's more
sample
basalt.
is
Right,
a
it's
rock
the,
and starboard
the
forward.
thing you get commonly
Starboard rock
with,
box,
with
right.
these types of seamounts is that manganese basically forms its own layer, and it may actually come off of the rock so that you end up collecting mostly just a slab of manganese. and not much attached to it.
So if you've just joined us, I'm Chris Kelly with the University of Hawaii at Manoa. I'm serving as the biologist lead for this particular cruise, and to my right is Jasper Conter, who's serving as the geologist lead for the cruise. So he's also at the University of Hawaii.
Is this a heavier or a lighter specimen? Is it going to float?
That's a good question. It could potentially float. I think I think you're in pretty good shape. The current's not too strong, is it?
That's enough to blow something away if it's a floater.
Boy, I don't know. I would suggest trying to drop it as far down the box as possible in case it is, but hopefully it'll settle down.
Oh, that didn't keep. Yeah, I'm going to try to get on the other side of the current, but not sure I'll get a good foothold.
We've got a different ROV crew, so maybe when they finish their collections, they could do a brief introduction of who they are. Around this time of the dive, we get people going in and out for lunch. And Jasper and I just took turns, and I think the ROV pilots are in that process as well.
Hydraulics are up.
Okay. All right. Let's see.
So while we are looking at this area, you can sort of see how the rocks that the sponge is on top of are kind of these bigger, rounder shapes. These are probably the manganese encrusted pillow lavas. And the smaller pieces in front are likely fragments and smaller pieces of pillow lava that came out at similar time frames. Some of them may be just fragments.
So we'll just go for the port side of the box.
Breaking off.
I'm not sure you'll be able to get the box open if there's a rock in front of my toe.
The actual smaller
give it
versions of the same
shot.
sort of pillow
Once
lava.
I have it. Yeah.
I might be able to port wing in, and that might give us the clearance
seems
necessary
particularly
to
that these harder surfaces
yep.
are the
Let me get it first, and then we'll
places where we see these
hydraulics
sponges.
coming on. Twitch.
How much of this are you planning on taking?
Probably pretty near the top.
let me know what cameras you need.
I'm pretty confident with this.
Okay.
So I'm going to get it near the top so that it's easy to get in the box, unless you need a good part of the stem.
No, we don't. That'd be fine, pilot. And then, pilot, what container are you going to place this in?
We're going to go for the port. Do the port inner.
Port inner. Okay, great. Thank you. I think we should just go as this is stocked sponge for the field ID right now. Maybe I can take a peek at the spicules this evening.
Zoom in, roll.
Do a little bit better job.
I can rotate as well.
Yeah, so this appears to be a soft sponge. You can kind of see the way it's wiggling there. And so that also is helpful. It narrows it down to couple of different families there.
Okay. Zoom out.
Want me to wing in first, maybe?
Yeah, let me get this clear.
Port wing inboard. Not all the way, but some of the way. And then as I extend the drawer,
I'll be ready to come off if I need to.
I'm watching Titan to see what happens.
Okay.
I can wing in further. Yeah.
Yep, you're hitting it.
Alright, yeah, we'll have to.
Yeah, I'll come up.
Yeah, so my best guess might be that this is in the family Euplectellidae. And it's in what's called the subfamily Bolosomini, which means it's stalked euplectellids.
What is that?
So, but again, we won't know
See,
until
like
we
the
look at
warm
the
fluid there?
microscopic parts.
I think that's a thermal climb of out of the bio box.
Okay. All right.
Yep. Ah, yes.
Yeah, I just.
Yep.
My mind went straight to hydraulic fluid.
Yeah.
But the comp is holding steady, so all right.
Okay, you are floating to starboard.
Okay. Let's try another look spot here. Just need the first minute here.
I'm gonna extend the drawer.
All right. Keep an eye on Sirius. I'm gonna bring up Mini Zeus here.
Okay. Drawer is open, ready to go.
Okay. And no tilt, of course.
I can help you with Titan too.
Yeah, I want to go help out. Why don't we put Titan up there?
We can do that.
Yeah.
It's zoomed all the way out.
Okay.
You happy? All is well. Yeah, I think your minute cam's going to be your friend here. Hopefully it's a sinker. Nice. Excellent.
All right, it didn't float. That was good.
It's clear.
Package is secure.
Good to hear.
I'll follow you with Titan if you want.
All right, that'd be good.
Very nice. Nice collection, pilot.
Thank you.
I'm gonna bring my camera back. I think we're stowed, I think.
Okay. All right, looks good.
Sorry, what was that?
Yeah, this is Charlie Wally.
Hello, Charlie.
Question is more about this sort of general landscape we've been looking at here. My expertise is shallow water aquaculture. This is a whole new world to me, but I've been struck by how many dead animals there are, remnants of them, especially the sponges, but some of the corals too. Much more than you would see on a typical shallow reef. And I'm curious whether that's sort of typical for these kinds of environments, or whether there's something going on.
Charlie, our interpretation when we see a lot of dead animals, which we do frequently, we see a lot of dead glass sponges on the bottom, as well as some dead coral branches, is that
we should tilt Mini Zeus
the
down
dead
a
parts
bit
actually
more.
persist for an extremely long period of time. So when the animal dies, it can just sit there on the bottom for many, many, many years. And in some cases, the actual dead sponge or coral stalks can acquire a manganese coating on them as well. So that means they had to have been there a
Co-pilot?
really long time. So I think the actual dead parts just persist longer.
Hey,
maybe
pilot.
to the
All right.
constancy
So
of the
everything's
environment down
stationary
here.
and settled out.
They're
Just
not
have a sponge
subjected
collection.
to some of the forces
We're
in
moving
shallower
up slope.
water that might
I believe it's roughly
degrade
330 is what we've been
the
moving
dead
at.
animals much more rapidly.
Oh, here comes Josh. Perfect.
So, Chris, is there anybody that would come and eat it, or is it just a matter of decomposition?
Excuse me, what was that again?
Is it a matter of something comes and eats it, or is it waiting for decomposition?
You know, I don't really know. For sponges, it's not really clear why they die. In some cases, you see these skeletons of sponges that had to have been extremely old. They're quite large, and so maybe there is a finite amount of time that an individual colony can survive. But you can also see smaller ones that have died, and you're not really clear why that is. Something knocked it over. You could get some of these fish or eels that might have bumped into it and taken it off its attachment, and then it's not going to survive very well laying on the ground. It needs to be upright in order to filter. Another thing that we saw last year, which has really intrigued a lot of us, is that we were in a place that had a lot of consolidated substrate as well as a lot of cobbles like we're seeing now. And animals on the consolidated substrate, you could get some very, very large ones that look like they're doing very, very well. And then you saw some other animals that clearly had been knocked over, and at their base, their attachment, there'd be a small cobble there, and our interpretation is that that particular coral or that sponge settled out on too small of a cobble, and that once it grew up large enough, it became top heavy for the rock that it was attached to, and you can get some really ripping currents down in deep water, and there might have been one of these internal waves or some very large current that just blew it over, and once it's fallen over, then its days are definitely numbered.
Copy. 30 meters, bearing 330, speed 0.2.
Sounds like a Wild West story down there.
Yeah, well, that's what we're trying to figure out what's going on here. You know, there's a lot of interactions between animals.
Hey, pilot, how do you
already
have me?
see, even in this type of community, animals living
Likewise.
on other animals, and the cutthroat eels that Bruce was describing too are just absolutely voracious predators down here, and so
The
they're very efficient, I think, at getting. I'm not really sure what they eat. Probably shrimp.
It's been
and
coming and
perhaps other
going
small
since the
fish,
dive
but I
started.
think
Has
they're-
it? Okay.
They're quite successful
Yeah.
predators. So a
And
lot
the
of
camera
predation going on.
actually
Sea stars
has the
eating
app
corals, sea
PTZ.
stars eating sponges, animals on other
That I was
animals.
referring to.
Oh, is the Manipcam new though?
It
Really
may be, I'm not
fascinating.
sure.
Okay.
I think it is
community
new.
in deep water.
I don't think it was going out before.
So you can see here sort of an example of what I was
the
talking
PTZ
about.
you're
Here you've
looking
got
at.
two
It's
big
not the Manip.
boulders, and now we've got a
Oh,
bunch
yeah.
of animals on it where we weren't seeing very much on those cobbles. You know, some of these corals could have settled out on these cobbles, but you know, they're preferring these larger, more stable type of attachment sites.
So just for clarification for the other
Copy.
geologists out there, when you say substrate, you're basically talking about the rocks that we like.
Yes, absolutely.
Is any of this interesting, Watchley? Like any zooms? So we're waiting for the ship to move at the moment.
So, Pilot, how about that kind of leggy, branchy one on the second boulder, the one behind the one that you're in front of, sort of on the right side of it on the top?
Yeah,
the lasers just went by it.
Yep, coming in.
It looks like it's different. So for those of you who didn't hear that,
try
you'll
restarting
see
that
the ROV pause periodically for a while, and in some cases it's just watch stand changes,
Come on in,
but
video.
in other cases it's the ship has to move. Okay, this.
I'm stable
What
if
do
you
you
wanna
think,
come in.
Asako? I'll let you take a stab at this first. I think I have an idea what this could be. So this is definitely an octocoral. And my best guess is that it's a bamboo coral. I think I see some black nodes on it. And also the bamboo corals, a number of species have the little
looks like
red
switching
or
it back,
pink
brought it back up.
oral
That's all you
disc
did?
right in the center of them. Oh, there was this shrimp that just took off. I wonder if we could look at a branch point of this coral, and that would help maybe inform. Okay, so this is definitely branching from the inner nodes. And so those little black spots or bands on the main stalk that's right in front of us are called nodes. And it's quite important in the taxonomy of this particular family as to where branches come off of the main stalk there, off of other stems as well. So in this case you can see it's coming off of the stem between the nodes, so it's called internodal branching. Other types of these bamboo corals, it comes right off of the node itself, and they call that nodal branching. So this is internodal, and it would put this into something perhaps Keratoisis or Keratoisidinae in that particular subfamily, although that's a really big subfamily. Asako agrees it's a bamboo. And this thing right next to it is a primnoid, kind of a pretty ratty little primnoid. Something bad happened to it. This is missing a fair amount of its colony there.
Yeah, let me know when you're all set, Watchley. We've got some more leash. Let me know when you're all set, Chris. We can move on. We have enough leash now.
Yeah, I'm good. Thank you, pilot.
All right, come on out, video. Thank you.
Go ahead.
Lesson 1, move complete.
Video switching out.
Well, we just started moving. How deep are we right now? 2,000? How do you feel, Sean?
We can wait maybe 2
Another one
minutes
of
and
these
put it in.
Corbitellinae,
I think that'd be fine.
New genus.
Do what Sean says.
And
Sounds good to me.
we've imaged a bunch of these already
The sponge,
today.
you're saying? Yeah.
So this seems to be one of the more dominant members of the sponge community down here. See, nothing really likes cobbles. So these animals are fussy, Jasper. They don't want just rocks or any rocks. They want big rocks.
Yeah, in
Let's do
that
a
sense,
quick
they're
zoom here.
kind of similar to geologists. Most of us like big rocks too. More rock means you have more to work with.
That's
Hold
true.
that. That's fine. All right, I'm just looking at sediment.
Looks like we're coming up on some bigger pieces here in the background.
Hey, Sean, can you try turning off the upper swing arms?
Turning them off?
The lights.
Yes.
Bottom left. Yeah, no. Keep them on. Yeah. Yep, we're just getting a lot of reflection here.
Yeah, Nav, if you want to put that move in, I think that'd be fine.
Go ahead, Nav.
So it looks like we're transitioning.
Ship move,
into one of
range
these
30 meters, bearing
larger
340
rock
degrees
areas
at
where
0.2 knots.
Larger pillow-like structures completely covered again in manganese. So we see the drape of manganese.
Come
on
in
some of
partial
these.
video. Other of these.
Yeah, I was going to suggest we look at that. I think it's a crinoid. Or maybe not.
Nav, bridge, move initiated.
No, what are you? Could be a sea star.
Alright, come on in video.
I think it's a crinoid. I think it's one of these feather stars.
Do you have any more?
Let's see if we've got some other
Oh, it's a
opinions.
little bit,
Yep,
yep.
we've got
That's
Barrett
cool.
weighing in on it being a crinoid. Asako thinks it's an asteroid. I think Sako, you could see the cirri from the view. That'll show up for you pretty soon. I think it's going to wind up being a crinoid. Yeah, there you are. So yeah, I couldn't tell whether it was a sea star or a crinoid either until they got close enough there.
Okay, I'll frame the guy.
So trying to identify these things from imagery is challenging. You know, I could throw out a guess here. It could be something in the family Antidonidae. I think is how you pronounce it. But that would be a pretty wild guess. But at least it's descriptive again, as we talked about. At least it describes the animal shape a little bit, and so that ID is viewed as more of a morphotype identification.
Let's do a quick zoom here. Probably already have seen this.
Holding partial.
Nav, watch lead. And the wrong one. Oh, there it goes. Yeah, Nav, could I get an update of how we're doing on our track? Roger, thank you.
Let's come in partial here.
Holding partial.
Yep, this looks like it's another one of these feather stars. 2, 4, 5, 6, 7, 8, 9, 10. I think it's got 10 arms. 7, 8, 9, 10, yeah.
When you're clear video.
So types of things that you look at in identifying those, of course, would be the number of arms. Again, some of these feather stars like to grab onto corals and sponges, and others like to hang onto rocks. And that's something large over there to the left. Not sure if it's something we've seen already or not. It'd be worth sort of taking a peek at it and seeing if it's something new.
I think you've seen this guy.
What is that?
Quick
Yeah, I
zoom.
didn't. Yeah, maybe we could take a look at this. This is okay. So this is another one of these stalked sponges. I don't think it's. I think this is more of a typical Caulophacus. It doesn't have that big opening on the top like the one we collected. So
all right, but that's about
that
70 in my camera.
should be good enough for
Come on
that.
up, Peter.
Color Fracas.
I gotta push ahead. See if we can get this big guy that was over here.
Roger.
So this may be just, pretty good size vase. Yeah, this would be perhaps worth taking a look at. And this is definitely different. This will be the first sponge that of this kind that we've seen on this particular dive.
Yeah, pilot, can I rotate maybe 5 degrees port, keep you in view?
You can just keep me where I am.
Copy.
Just keep an eye on that slope.
Yeah.
Come on in, video. And I'm going to approach it as we hold partial.
Roger.
Okay, this is great. This is a very interesting sponge. And I've got to remember what we think this is. I've got to consult my little cheat sheet here. My cheat sheet is the Office of Ocean Exploration and Research's online deep sea animal guide.
Lost that camera again.
So it's a very useful thing for those of you who are interested in identifying
Hey,
deep-water
video,
animals. That guide
when you
has
get a
got
chance,
1,700 images in it.
Switch the.
Oh, it's
the
a
nib cam
really
you just
wonderful
lost.
reference.
He switched it off and then back on. I think just
I'm
screens.
going to take
Roger.
advantage of right now.
That seemed to be a temporary fix at least.
Could we turn off the lasers for this one?
Yep.
Yes.
Okay, come on in, video, when you're good.
Roger.
Looks like it's well over 10 centimeters across. This is a pretty large
Lasers off.
feature here.
Roger, Nav, go ahead.
Nice.
Yeah, pilot, not too much longer.
Okay, great. Thank you, Nav.
Coming out just a touch.
Nav, bridge move complete.
Okay, I'm gonna come out for a establishing shot.
It has got to be in this group.
Yep,
Where
maybe just a
are
few more
you?
seconds, pilot. You're starting to get back there.
Let's see here. Okay, I think
Copy that, Nav.
it's got to be in the family Rosellity. Let's see if I can.
Okay, coming full. We're already full way. Great.
Okay, I'm getting closer here. Okay, well, it could be something that's called
see it.
Lophocalyx, but I think for sure it's going to be in the family Rosellity. And so that's good. We, I don't think we've seen any of those in the Marianas cruises that were just completed. And I don't know the name of this one very well because this was a new one that we collected in the northwestern Hawaiian Islands. It was new to me, and it actually hasn't been really examined carefully by one of the world's authorities. There's a couple of them that are world's authorities on the deepwater sponges, so it's tentatively Lophocalyx based on some photographs I sent them. This is a pretty interesting sponge. I wonder if we could get a look at this one.
The one in the center?
Yeah, this very large, globby one on the side of the rock.
Yeah, can we get lasers on again?
Yes, lasers are on.
It's like the animals are picking up a bit too. So this looks a little bit like this one sponge that had a bunch of commensal anemones in it from last year.
Okay, come on in, video.
Holding partial.
Okay, so it does not have the anemones with it. We would see them by now. So I think it's safe to say that this is in the family Eurydidae, or something. Yeah, I think that's the correct family, and
Could we turn the
this
lasers
could be
off?
something in genus Heterorhitae is one possibility.
Go ahead. Go in.
There's also genus Perifragella as well that it could possibly be. So the interesting thing about this is a glass sponge, as all of them have been so far on this dive. And this is very, very brittle. And some deepwater sponges are very soft and sort of flexible. This one, on the other hand, is very brittle. So if you were to grab part of it, it would just snap right off. And sponges get their brittleness, if you will, by whether or not the skeletal elements, the spicules, fuse together. And if they fuse together, they can form a very rigid skeleton. If they don't fuse together, then they can have a very soft, loose type of skeleton that's flexible, and you can squeeze it and so forth. And so that vase sponge that we were just at a few minutes ago, that was a soft sponge, whereas this one is a very rigid sponge. You can actually see the sort of cross-hatching in the sponge texture here, and that's the skeleton itself. It's called a dictyonal framework, and that framework is created again by the elongate skeletal parts actually gluing together and forming a rigid matrix.
Full organism shot.
So Chris, is the dark stuff just sediment getting caught in this, or is it slowly dying off?
Yeah, it's dying. So oftentimes you'll see parts that are dead on the sponge, and then you have a whole living portion as well.
Chris, is this a separate sponge, or is that part of the same species?
Can you say again, Pilot?
Is this a separate sponge, this part, or is that part of the same sponge?
No, I think it's part of the same sponge. Yeah, I think it's the same, and it's just this brown part it's just died for whatever reason.
All right. Are we good with this?
Yeah, this is fine. Thank you.
All right. Come on up, video. Coming up.
Full wide.
Thank you.
Another one of those vase sponges. Some more of these Narella. It's an interesting sponge in the back there that might be worth taking a look.
Which sponge is that?
Yeah, that one in the back, that sort of large one that looks sort of like a tree.
Ah, okay.
And the one even below the tree, that looks different as well. Like they're two.
You're going off to my starboard. Can I rotate a little?
That we haven't imaged before.
I'm going to end up right in front of you.
Okay.
I keep getting pushed to my starboard.
Copy.
By the current, so.
All right, I see that we've been joined by Tina Molodtsova. She's from the Shirshov Institute of Oceanology in Moscow, but she happens to be in the Netherlands right now, so welcome, Tina. And she is an absolute expert on a lot of animals, and also in particular black corals and tube anemones and all types of things. These anthomassus, these alcyanation or soft corals. So delighted to see that she's been able to join us, even though she's on travel right now. So this particular sponge, I think, is in the family Farreidae, this very tall, sort of skeleton-looking sponge, and I think it's in the genus Spidoscopulia. This
you can zoom as you wish.
I was misidentifying these sponges because I was not aware of this genus for quite a few years until Tina had actually gotten me in touch with one of the two world's authorities on these glass sponges, who's also at the Shirshov Institute of Oceanology in Moscow. His name is Konstantin Tabaknik, and he actually described this genus, and so he really knows these things well, and he sent me a paper, and I started realizing that we were seeing a lot of these things, and I was misidentifying them.
I'll come out slightly wider.
So one of the real values of this type of a cruise and through telepresence is that we get a chance to interact with specialists and experts around the world. And as Tina is showing out right now, she's showing us right now that you can participate from anywhere in the world as well, as long as you've got an internet connection and a laptop or an iPad or something.
Okay, yeah, I see.
Oops, okay, so Tina is not in the Netherlands. Did I blow it? Okay.
Let's come out and get the base.
I see, so Asako is in the Netherlands. Excuse me, she's not in Chiba, if I'm understanding the chat correctly. So maybe Tina is in Moscow. You could see a variety of shrimp on these sponges. Again, reiterating the point.
Okay, we can get a full organism.
These types of animals serve as hosts for a variety of other animals.
Lasers on.
And now Tina says that she is indeed, she's not in the Netherlands, she's in Newfoundland.
Yeah, we can use lasers.
So again,
kill lasers?
It's pretty amazing. We've
Lasers
got people
off.
now participating from all different places here in the world.
Do we want to look at this other sponge?
I don't think so. I think it's the same as the one we imaged. It just doesn't have any dead parts. So I think we could probably move on.
Copy.
Thank you.
Do we have a ship mover?
I was just about to ask.
Yeah,
now that we're done with that. Great. Yeah, 350 looks good. Does that put us on track to... Yeah, I guess we're pretty much going north. Yeah, 350 looks good.
Go ahead. Ship move, range 30 meters, bearing 350 degrees, speed 0.2.
Charlie Wall has just shared my enthusiasm for sponges, calling them spectacular animals. Yeah, I totally agree, Charlie. I used to be a fish guy and then wound up doing some work on some sponges and have been hooked ever since. They're really amazing once you get to know them. So, Pilot, are we looking at a dead skeleton here, or is it? You can't seem to see the polyps there. I wonder if that's worth a zoom.
Yeah, we can take a zoom.
Okay, thank you. Yeah, starting to see them now a little bit.
Yeah, come on in, video.
Holding partial.
Okay, this is a primnoid coral with some brittle stars on it. Yeah, that's also, Tina also agrees. My guess would be probably a Norella, but it's certainly not certain.
Then pan back down
you
to
might
little
bump.
stars.
You're drifting slowly towards.
What did I say?
Yeah,
I
you
guess
just
my
bumped a little.
guess would be. Oh, that's great.
That's double guessing.
Double guessing, yeah.
That's better than second guessing.
If we have time, could we pan down to the brittle stars again?
And that sponge is different too. Could we get a zoom on that sponge when we finish with the coral here?
Absolutely.
Because I think that's the first time we've seen this sponge.
And someone's hugging
Yeah, you got a little brittle star who's got his arms wrapped around. I think he's on the other side. You got a real star on the coral, and also these little white arms here on of a brittle star's body, I assume, is on the other side.
I gotta reposition a little bit here.
No worries. Come out.
And then
Yeah, you have. Yeah, you see it.
I think that sponge could be something in the genus Lefroyella, but I'm going to look and see if we can see these anemones that are associated with this genus. They have a commensal relationship with the sponge. And I thought I saw them. And I guess I'm wrong. Looks like there's something else there, just some debris on the sponge.
Yeah. A bit more zoom.
Yep, go ahead in.
That's full.
I'm going to put this down as Lefroyella species question mark, even though it's missing the anemones. It certainly looks like it. Anyway, it's different than any of the other ones. So again, we have yet another species of sponge down here that we just documented.
All right, I'll get a quick establishing.
Well, it's whatever on
All
depth
right,
is.
full wide.
We need to be at 50 meters at 1630. So you have to do the calculation to figure out when we have to come off bottom to do that. Yeah. Okay. Top center. Yeah. No, we're not making that. I'd like to.
I think these.
So let's keep the ship moving.
So this is a big Poliopogon sponge. There on the bottom right in the center. We've seen a couple smaller species in that group. This is, well, I don't know if it's Poliopogon now. No,
Yeah,
I
when
think
was
it's
that last
actually the
news put
same
in?
one that we imaged before, but it's just a really good sized colony.
Okay.
Sure. Yep.
Yeah.
Let's come in partial. Let's get the lasers on.
Yeah.
Lasers
Go ahead.
are
Yep.
on.
$30 meters at 350 degrees at 0.2 knots.
So this is a very delicate, brittle sponge here, and again, probably in the family Eurydidae, but I'm not positive. Anyway, an absolutely
Nav grid
gorgeous
emitted.
animal, as you can see. Very complex
Lasers
morphology.
are off.
And again, this could be something in the genus Heterorhitae, but I'm not positive.
Do you have any more zoom?
Yeah.
Yeah, come on in.
I'll just hold a partial here. Get
So
the
I'll just
layers.
sort of throw this out. Who knows whether it's right or
Sort of
not, but
petals.
oftentimes animals will have all of these sort of leafy complex structures in an effort to increase surface
just wanna
area
sort of
that they're
pan
exposing to
around
the
it.
water. And it's common in filter feeders in particular. As I mentioned before, the water is going to be coming into the sponge here on the side, and on this particular type of morphology, it's not really clear where it's exiting or or what the path of the water is in one of these sponges, but it's very likely that this, again, this very complex leafy structure is to provide maximum exposure to the water and to improve filtering efficiency.
You want to come out just a little bit? Okay, I'm going to move on.
Roger. I'll hold here.
And as far as aging these things, boy, I don't know. Nobody knows right now. Trying to age a sponge like this is extremely difficult.
Coming away. Nice.
So do some of the more linear looking or flat looking rocks there may have actually been these little lava flows we call sheet flows. You get underwater eruptions that are either pillow lavas, that are these sort of balloon-like structures that basically slowly outpouring melt, freezes into cold water into these little balloons. But if there's enough melt coming out at the same time, it ends up making these little lava flows, usually pretty thin. That's why we call them sheet lava flows. And so some of these longer, flatter looking rock formations might be part of that.
Any interest in any of these corals?
No, I think we've imaged all of these, pilot.
Yep. Continuing on.
One on the right looks like Candidella gigania. And here's a dead sponge in front of us. I think somebody might have joined the line. Is that correct? Okay. Sort of a tall sponge there, which is possibly in the genus Tritopleura. It's dead. I think we saw one of these that was alive earlier. I think I've recorded it here, I'm pretty sure, yeah. And we got another dead one of these Spidoscopulia as well. That's the really branching skeleton type sponge on the left. Looks like we're getting into a little more consolidated substrate again, so it's kind of like alternating between.
Snap zoom on this.
Yeah, that's kind of what I was noticing too. We seem to sort of hit these pockets that are basically the broken down pieces in between these larger rock formations that are leftover bits of lava, of mostly big pillows, but some of these sheet flows as well. And then these pockets have smaller rocks with sand.
When you a great video.
There.
So that animal was sea cucumber or holothuroidean. As you can tell, they look quite a bit different than their shallow water
bridge move complete.
Varieties, and
yeah,
that
pilot,
one
if you're
maybe was in the
we
family
could,
Synallactidae. I'm not really sure.
Right now, if you can, yeah, same thing. I think is fine.
Can wonder if
Go
we
ahead
can
now.
take a zoom on that sponge there. I think this is possible Farreid.
30 meters at 350 degrees at 0.29.
A lot of different types of sponges today. You know, some corals, but definitely a larger variety of sponges at this location.
Now, bridge move initiated.
Okay, and this is something that we call Faria near Oca erecta, and basically it's in the genus Faria in the family Fariidae, and there's a bushy form or we just call Faria near Oca. Near Oca means that it hasn't been conclusively determined that it is the species called Faria oca, so we call them Faria near Oca. And that's the bushy form, and then you have these sort of elongate forms that come upright, and we call them Faria near Oca erecta because they're very erect colonies. This is a very brittle sponge, and it's good that we're able to document this because that's yet another different one that we're just seeing for the first time today.
So Sean, the little piloting tip.
Interesting looking with the little almost tube-like structures that you get here.
Watching my toe, and just with the corner of my eye.
And again, my assumption is that the water's coming in the side of the sponge and going out those tube structures, out the mouth of those tubes. But it's a very tortured route. I mean, the tubes are going in all directions.
Because of that.
Yeah.
So it maximizes potential to catch things, right?
Yeah. But you can also see how thin-walled this particular sponge is too, so maybe it doesn't even matter in what direction the water comes in. It's not really clear.
It might not be obvious what's going on.
Faria oca is actually known to have only just a few layers of the framework. And this is another one of these brittle sponges that has a framework in them.
Slightly to starboard.
So the water is going to pass through it really quickly. If you're just joining us, this is a live broadcast from the NOAA ship Okeanos Explorer. We are currently on Alba Seamount, which is located about halfway between Guam and Wake in the western Pacific Ocean. Our depth is about 2,124 meters, and we're at a temperature of about 2 degrees Celsius, and our dissolved oxygen is around 3 milligrams per liter.
Skimming partial here.
We'd like to welcome everybody who's watching the broadcast today. And here is a beautiful stalked crinoid.
Holding.
I'm waiting to see if we get a little help on these. There are not that many crinoid folks that are out there, specialists.
Got a bit more soon.
This particular one, to me, looks like it's in the family Hyocrinidae, but I'm not absolutely sure. Hyocrinids are known to have 5 arms. This one has 5 main arms. Oh, wait a second. Can you zoom in right below the arms? It's got the discs. Wow, this has got the discs.
Yep. Wow, that's full wide.
So we collected one of these things in the northwestern Hawaiian Islands that's a little bit different than this last year. And it's a suspected brand new genus of stalked crinoid. And the one thing that the specialists were looking at were these very circular discs. And
you're talking about
so
these
Tina,
little tiny
and so Tina Molitsova, who's on, she's suggesting collecting this animal, and I would agree with her. I don't know if anyone else wants to weigh in on this. This is an extremely unusual stalked crinoid.
Yeah, can you back them up to like
Maybe
10 meters?
Yeah, actually.
Stopping move, coming to position.
Yeah, so...
Apologies, Nav. It's very loud up here. You want to go 10 meters in the reciprocal course, correct?
We collected in the northwestern Hawaiian Islands.
I'd go for it.
170. 10 meters at 170 at 0.2 knot.
So we have a sample in the port inner, and I believe that's the only bio sample.
Yeah, so I'm actually looking at the picture of the one we collected in the Northwest Hawaiian Islands.
Bridge move initiated,
and
headed
it is
10 meters,
This
bearing
one is different. The
17
one we
degrees.
collected in Northwest Hawaiian Islands did not have the very fine filaments coming off of the arms, whereas this one does.
Can we come full wide?
Yep.
But it may be in the same new genus because of those circular discs
Spell
right
my
below
ports
the
when I-
calyx.
Absolutely.
Yeah, and Tina is also commenting that it's different as well. So this could be a very valuable collection here. Tina, I wonder if you could put in the chat the name of the expert there. I've forgotten his name. Who's the crinoid expert in Moscow who works on these sea lilies?
Yeah, he's good spot.
Yeah, so she's come back. His name is Alexander Mironov. And so he was also extremely interested in this crinoid too. So we may actually have two different species of a brand new genus.
Copy.
If you want to get that
So
box ready.
what's in the
Yeah,
port
do
inboard?
you want to go to starboard?
There shouldn't be anything in there, right?
I think that's where the last biosample went.
Yeah, all right, excuse me. Port inboard is right where the bio went. And then we got the rock box, right? It's got the rock, and that's it. Right? Okay. Thank you.
You move my utility camera down. Yeah, that's not.
So you want to
that's
get
not
the.
helpful.
Yeah.
Is this a good spot to grab it, Watchley?
Yeah, that's great. This is the only second time we've ever seen a stock crinoid that's anything like this. Those very, very unusual round discs underneath the top of it on the stem. That's what we were looking at. So it's pretty exciting to encounter what is very likely a brand new species to science.
Yeah, Sean, you need to pull up.
Copy.
So, this is going in port, starboard inner.
Starboard inner, okay.
Yeah. I've got to move a little bit because Sirius got way ahead. So, can you drop a waypoint here, Nick? You did? Okay. So I'm going to move. And then we're going to stow this.
Yeah. The other one closed up too. That's the same picture. But it's a different arrangement of the discs right under the calyx, and again, it didn't have those other projections coming off of it. Whoops. You can't see those. I don't know. I think of them as hairs or something. I don't know what they're called. Yeah, but that's okay. It's not going to be very far away from that, and it's better to make sure it's actually in the box.
Yeah, and we marked the Latin long on HiPack, so we can go back and correct that in the log if you want, Chris.
I think you're really not moving that far away, so I think it's fine. So is that just manganese crusting over sediment or something? That's
Right,
not like
Ready?
a sheet flow, right?
And then, yep. I gotta try a different approach here. Can I get the mini Zeus pointed down? Or actually you can't.
Oh, that's right.
This is being very difficult. Sorry for the time this is taking, watch lead.
Yeah, no problem. I fully understand. I think the specimen is worth it, though. It's really.
Is the specimen on the other side of floater?
I think it settled down okay, didn't it?
What's that?
I believe it sank, but it was dropped in.
Okay, let's try the other side.
Copy that.
I'm having a lot of trouble here.
Yeah, closing drawer.
So the tricky part for our viewers about this is that some of these animals just lay right down. You drop them in the box and they settle right down, and other ones can tend to be neutrally buoyant, and so they just go off in the current and you lose them. So you have to be pretty careful. And we've got a fish.
Come down with the manip, the utility cam, please.
Absolutely.
That's good. I just want to make sure I'm not going to hit the shilling.
Oh, yeah.
This is Bruce again. This is another one of these cutthroat eels, the Synaphobranchus species. I've had some communications with Ken Sulak, who's an expert on this group, who's watching the videos now. He's trying to log into the chat room, but is having difficulty. I know you can't do it now, but he's pointed out for these eels, it's useful to get tight zooms on the head and on the body just behind the head, because the nature of the pores on the around the head, and then also the scale pattern, if you can see that, is very useful for identifying these species. So we just saw another cutthroat eel in the genus Synaphobranchus.
It's in there. It's port outer.
This is great. Thanks, pilot. I wonder if we could take a look at this eel before we head out. Just
absolutely. Come on in, video.
Holding partial.
Oh, there's an urchin he's about to go over too. It's like it's Spidodiadema.
It's taking a bit more zoom. Okay, I'm going to zoom on the head.
Bruce, has your image caught up already? It looks to me like the dorsal origin is pretty far back.
Yeah, we've changed our system here now, so, and actually everything's working really well. So it looks like it's pretty far back. Ken pointed out there are actually two species that have a dorsal fin that far back. The one that we've been calling out, Synaphobranchus brevidorsalis. There's another one, Synaphobranchus oregoni, that also has a relatively short dorsal fin, maybe not quite as short as brevidorsalis, but there is overlap in that character. So he was asking, for those close-ups of the head and the body. I just got some nice zooms on that. We might be able to see pores on the head, and maybe he can do something with that. I'm not sure that you actually see the scales.
Yes, lasers on.
When they're alive, but he seems to be confident that with good zooms you might be able to. So we'll see. I know he's watching. He's been trying to log into the chat room, but he hasn't had success right now. He's had some problems. So it's either Synaphobranchus brevidorsalis or Synaphobranchus oregoni.
Video.
Okay, thank you, Bruce.
Zooming on the head.
It's interesting to see how scarred so many of these
Lasers
cutthroat eels are. They're active predators. They feed on shrimps and cephalopods and crustaceans. They're one of the few deep-sea fish that are energetic enough to actually do a lunging attack at their prey, and I'm wondering if some of these scars and things, particularly around the head and snout, are for lunging to capture prey in among the rocks where they're getting kind of scraped up. There may also be some interactions with other species or members of their own species, but we do see a lot of eels with these white
let him
scars.
swim out of the frame.
Yeah, Bruce, I've seen some attacks by amberjacks on fish, and if these eels are anything like the amberjacks just blasted in and just went after their prey, just smashing into the sediment. Or the substrate. They didn't even care. And it's possible these eels are kind of the same way. When they actually got a prey in sight, they go for it, and they don't really care if they get banged up a bit.
Yeah, I think that's exactly it. I think that's what's going on. We'll see if Ken has something to say about this a little later, but I think you're onto that, Chris.
Is this sponge of interest?
And those are great close-ups too. Thanks, camera operators, for getting those zooms on the eels. That was really wonderful.
Sure, thank you, Bruce. Let's see. I think we've imaged this before.
Let's do a partial video. It's quick.
Yeah, we could do that. I think it had a stalk. This one, which is a little bit different. Yeah, he's got a stalk.
Pilot, do you want your port lower back out?
Clear.
Yeah, you can deploy that.
So it's... okay.
Yeah, we can start moving again.
Copy that. Nav, we could probably do the same.
So also, Jasper, I guess you
Same
want to
thing.
keep in mind
That's fine.
where second rock collection, if you see something particularly juicy.
Now, I was hoping this time to pick up one that's a little bit more angular from one of the more solid areas. Just not so solid that we can't get it, but...
Right. Another one of those dead sponges. So who knows how long their skeletons can just sit there on the bottom. We just have no idea. But I mentioned earlier that we've seen some of these stocks that have a manganese crusting on them, and that sure implies that they've been down there for a while.
It seems all of us
ahead,
got
Nav.
booted out of the event log for a little bit, but most people seem to be getting back on again.
Roger, copy. Bearing 3, range 30, bearing 340, 02 knots.
Good copy. Thanks, Rich.
Copy that, Nav. Yeah, pilot, it looks like slope's starting to change actually to starboard. So are you comfortable to going it at a slight angle towards waypoint 2? Or do you want to go upslope?
I prefer to go upslope, but this isn't too bad. It's pretty
Okay.
steep though.
Yeah.
Just gotta keep an eye on your sonar.
Copy.
You might want to keep your head directly into the slope if you can.
Yeah, it's only a slight rotation.
Move is initiated.
Another one of these vase corbitellini is what I'm calling them for the moment. A lot of these sponges can have very different shapes. I think their growth patterns are dependent upon current and where they settle out. So they can kind of fool you. You got to look at specifically at certain features to have a shot at trying to identify them correctly. And even then, it's challenging.
A long whip.
Yeah, there's a long whip. Maybe we could take a look at that. It looks a little different than the other whips that we've encountered.
Just gonna shoot over there.
A little
real quick,
Caliphacus sponge
Yeah,
down
absolutely.
below it. And an Norella, I think, up
I've got
above
40,
it.
so you have
One
time.
of these primnoid octocorals. So these bamboos are a real problem
Come on in, video.
taxonomically. There's a couple people working on this family of octocorals, and very complicated what they're finding out and what the genetics are telling them. So this has been an ongoing project for quite a few years, and it's still very confusing, but I think they're fairly close to
I'll just take
publishing
a bit more zoom.
a complete revision of this family. Hopefully that'll come out in the next year or two. So for the moment, at least, I think we're going to go with Lepidopsis. They're unbranched versions of these bamboo or isidid corals, and they also tend to have what they call intertentacular spines that you can see on this particular animal.
Gonna take a tiny bit more.
Got a squat lobster there too in the background. And it doesn't have very many sclerites or rods in the actual stock, so hopefully that'll be informative.
It's definitely something there. It's hard to tell what it is.
Know what that is, some type of a gelatinous thing. Maybe it caught some type of an old larvation house or something like that that was drifting.
Yeah, can't tell at all.
Yeah, I know, or a brittle star or something, but who knows what that is. Yeah, so it definitely is lacking a lot of the sclerites in the column, so that'll be very informative when, again,
Pilot,
when
I'm starting
some of the
to
experts
lose you
get a
unless
chance
I
to look
rotate
at
a little
it.
more.
You gotta come out. Video.
Roger.
You're just on the border
Away.
there.
Coming up, heading to port.
There's some serious manganese crusting on that rock. Look at that.
But nicely, you see these nice big blobby shapes sort of draped inside of it. It's probably a little mound of pillow lavas.
Now this is bridge. Maneuver is complete.
Copy that. We can get recentered and then probably put another one in. Give me 1 second. So this is me pointing due north.
Yeah, you can turn a little to port.
Yeah, how about 350? How do you feel about that now?
It's fine. Fine with me.
We've got another fish, probably another one of these cutthroat eels.
Fish.
Where's the fish?
The
To
fish
the
is to
left
the
of that
left
boulder.
center.
Ah.
The third in, yeah.
Okay, come on in video.
Yeah, Nav, you can go ahead and
Hold
put that in.
Go ahead now. Roger, copy.
So
30
Bruce, the
meters.
dorsal looks pretty far back
350.
again, but I don't know if you're
0.2
seeing
knots.
this yet, or you could concur.
Yep, I can see it, and I concur. And this one looks like it might be too far away to really zoom in, so we'll have to just kind of guess that it might be the same species that we were seeing before. And again,
Come in, take.
Synaphobranchus brevidorsalis is the one with the really short dorsal fin, but there is this other species, Synaphobranchus oregoni, that also has a relatively short dorsal fin. Oregoni much more poorly known, I think, than brevidorsalis. It has been recorded off Japan, and I think in the Kyushu Palau Ridge area. So it's thought to have a circumglobal distribution, just like Synaphobranchus brevidorsalis. We'll see what we can do with these. Yeah, thanks for zooming in. That's great. You're doing a great job with this.
Oh, sure. Yeah, you're welcome, Bruce.
Is that good, Chris?
Yeah, that's good. Thank you, pilot.
Yeah, when you play your video.
So could we use the scratching pattern, the scars on his back, to see if this is the same guy as the one we saw earlier?
Yeah, he's not. I think his head didn't have the scratches on it. But
yeah, that's an interesting observation. People use that sort of thing to identify whales, scars and scratches and blotches and all. So you could probably, and they also use it for sharks. So the reef, white-tip reef sharks, for example, there was a project here in Hawaii to try to track individuals using the markings on the body. Unfortunately, we're not going to get the intensity of observations probably to see the same individuals over and over again. So the deep sea exploration is kind of a one-shot look at each individual, and we'd be extremely lucky to see the same individual more than once. Having said that, there were a couple of places in the last expedition's leg where we did see some of these cutthroat eels over and over again around sites, some of the hydrothermal vent sites where we sat for a while. And looking at the images to look at those scarring patterns could tell us if we were seeing more than one coming into the ROV or if we were seeing the same animals around and around the ROV. So it's an interesting thought.
yeah, one of these days, perhaps there can be some bait mounted on the ROV itself. And I think you've made the suggestion, and others have made the suggestion as well, that you could probably increase the number of fish that are encountered by the ROV by just having an attractant, shall we say, to it, and get a better idea of just how many fish are in the immediate area.
Yeah, that's true, and we might also see some other animals like the very large species of amphipods that come to baits in the deep ocean that we aren't seeing so far because those things probably stay buried in the sediments until a food source arrives for them.
Yeah. As we're going by these rocks, we've got some of the same crinoids, these feather stars that we've imaged before, and more primnoid corals, a little vase sponge. Again, so we're seeing a lot of the things that we've already imaged. And so we've typically, when we start these dives, you see a whole bunch of different things, and then it tapers off. You eventually start getting, you've encountered many of the animals in this immediate area where the dive's taking place that exist there. So we're getting to that point. We've got another one. And I wonder if this is yet a different one. It looks like it definitely has a different scarring pattern on him. Looks a little smaller too, thinner.
Holding partial.
Okay, this is another cutthroat eel, and now the question is, does it have the same scarring pattern as what we just imaged before? Is it the same individual? And the answer looks like it's no. This has got a completely different pattern of blotches on the side, light-colored patches. So another cutthroat eel, Synaphobranchus, I'm sure it's probably the same species as the one we've seen about 3 times before now. If we're close enough to get any zooms are useful if there's time, but if not, then that's okay.
Yeah, so this is definitely different than the ones that we just imaged that clearly had scratches on the side and didn't have the blotches. So it may very well be brevidorsalis, but it's certainly not the same individual. So there seems to be a fair number of these cutthroat eels are in this area. It makes you wonder what they're all eating down
bridge
there. We haven't
maneuver
seen
complete.
tons of shrimp and tons of things. So I don't know.
Yeah, we're not seeing any little fishes that they might feed on. We haven't seen any small fishes at all. So, yeah, it's a good question. You know, what's going on? What's their food source?
Do you have any more?
That's full.
That's full. All right. All right. Just let him swim out.
Roger.
Oh, thank you for the zoom on that.
You're welcome,
Yeah,
Bruce.
now go ahead.
Thank you.
Pilot, are you okay
I
with
see
that? I
that
have you at
Patty
45
Fryer
now.
has joined us. Welcome.
Push
Hello,
up a little
Patty.
bit.
She was the, as many of you recall, she was
Like 2 minutes. We
the
can put another minute.
geology science lead, onboard science lead for the last
Then it looks like, for me, it
expedition
looks
in the
like
Marianas.
upslope is about 340 now.
In addition to being really, really interested in the Mariana Trench and mud volcanoes and all this other stuff, she's also interested in these older seamounts like this Guillote that we're on. Okay, Patty, we want to do a little exercise with you here. So Jasper pronounces that type of seamount a little differently than I do. So we're going to go ahead and each say what we think the pronunciation should be. And then in the chat room, you can say who's more correct, okay? It'll be a little friendly onboard competition. So I'll go first. Correct, okay? It'll be a little friendly onboard competition. So I'll go first. And this is Chris. And I say Guillote. And now Jasper.
And I normally say Guyot. But then the question is, who's closer in pronunciation? Yes.
Do a quick zoom.
Oh, but who win? You say Guillote, but
Guillote?
Yeah.
Or should we say Geo?
She said, is it Guillote or Geo? Guillote, I think.
I know you don't say G. It's a G, but the way she's written it here might be Geo.
Right. As we're discussing pronunciation, this is a Narella, one of the primnoid corals, a really beautiful specimen.
Yeah, we're near clear video.
Maybe a little bit different than some of the other Narellas that we've seen. That was a.
Did you want a close zoom on that, Chris?
I don't think so. I think that should work out.
And Patty declares Chris the winner.
Oh yeah. I'm not competitive, am I?
No,
not at all. And the only reason I know this is because Patty was almost pulling her hair out in the ECC one time while I was trying to learn the correct pronunciation.
Go ahead, Rick.
It's like taking your fingernails on a chalkboard the way I was saying it. It was torturing her. Another
30 meters, bearing 340 degrees at 0.2 knots.
And it's got some zoanthids on it.
So, pilot, when you see a rock in
Now,
this
bridge
area
movement
or
initiative.
something similar to this that we might pick up.
Yeah, copy that. Yeah, I'll try to look for something loose. But just let me know if you see anything in particular. Can you get lasers on, Sean?
Yes.
It's hard for me to see how close you're going to be able to get to any of these.
Well, you tell me what you want, and I'll let you know if it's possible to get in there.
Okay.
Yeah.
Yeah, I think that you may want to clear the bigger ones here and then look in the next patch here.
Okay. Yeah. Or, well, hold on. We're going to move a little bit and see if we can find a loose one. Can you fire up hydraulics, Sean?
Yeah. Hydraulics up.
Maybe something in this pile right here, Jasper?
Yeah,
that's
a little
awfully
up
big.
to the left. Up to the left a little bit more, maybe.
Boy, that's a whopper, though, isn't it? Or is that too big?
This is Maybe a little to your right. Yeah, that one of those two maybe.
You're carrying it up the stairs. I'm telling you right
A
now.
little big.
Let's see. That is, that's 20 centimeters across.
Yeah.
Oh, the tumbling snail. Look at this. It's over on the left, right on the upper left. Could we just detour just very briefly? These are the genus Gaza.
Come on in.
You saw them in the Northwest Hawaiian Islands. And Mary Wickstein has got a paper out on this. She submitted the paper from the Northwest Hawaiian Islands. Oh, poop. Come on, keep tumbling.
Swimming tight.
He knows we're looking, so he stopped.
Yeah.
Pilot, do you want that easy stop while we...
So that's great. We haven't seen very much of those. I think we had 1 or 2 encounters last year of these snails, and none so far in the Marianas this year. So.
All right.
Yeah, we can stop. I'm going to poke some of these with my toe and see if they're loose.
There's a dead, looks like sponge stock there. So again, that's just really common at these depths where you see these dead stalks.
Go ahead.
What about that one there? Or that one?
Yeah. I think that's...
Stopping the ship.
Oh, okay.
Which one are you guys looking at? Jasper or Chris?
What was, excuse me again, pilot?
Which rock are you looking at?
Is
just a little bit to your right and a little down. A little more. Yeah, laser just got on it.
Okay.
That one or the one where the laser's
Or-
next to
or
it
the
now?
one, yeah,
Either one of those?
The one next to that
Yeah.
big rock to your right,
Yeah.
bottom too.
Copy. You got hydraulics, John?
Yeah, hydraulics ramped up.
All right, the real test. Will it move? And the answer is...
No, it's probably stuck.
That one's stuck pretty good. Hey, there, that one moves.
Is that of interest?
What do you think about that one, Jasper?
Is that guy or
It's pretty good, huh?
that guy's loose?
All right, you got a selection
Yeah, let's look at that first one.
here.
It looks a little bigger, a little more angular.
Pilot, where do you want to put this one?
Well, it's going to have to go in the port.
Okay.
Okay, so port rock sample 2.
So let's take a look at it.
Copy.
Can you tilt my camera up?
Absolutely.
Roger, Nav, go ahead.
Tilting up. Stopped.
Yeah, come on in.
Great, thank you, Nav. And this
Covered
will
up slightly.
complete our collections. So
Stopped.
hopefully we'll be able to make that distance, perhaps. So what do you think?
That looks like a
Swimming just
sedimentary
a bit
rock.
more.
Great, thank you.
Can we turn it back to that angle you had initially? Yeah. That looks kind of like a sediment to me.
Go ahead and stow.
Is that no good?
That depends on what kind of geology you do.
Is this the type of geology that you do?
This is not the kind that I do.
So you don't want it?
I think that one is sediment with manganese on top.
Tilting down.
Did you want to try for another one here, or do you want to move on to a different spot?
I think we might as well move on. I think that we're going
Oh.
to find... Yeah, see, that's,
Yeah.
that's sort of...
Little attitude in that drop.
I think that seals that one as sediment.
Alright, so move on.
Yeah, let's find something better.
Alright, copy. Alright, let's get a ship move in.
Yeah, I don't see much on sonar, so I think that's fine. 340.
Go ahead. meters at 340 degrees at 0.2 knots. Now, bridge movement initiated.
Quick zoom on this crinoid.
Yeah, so that's a different one. This is one of these, what are they called? Cerametra, I think. I gotta go look that one up. These beautiful purple comatulades, and you see how it's attached to the sponge stock there. That's what's called prehensile cirri, and that they can actually do several wraps of their cirri, or the little feet there at the bottom of them, around stock. And not all crinoids can do that. So that's definitely one of the features, again, you look at when you're trying to identify these particular animals.
Do you have any more zoom?
Yep.
Is that a worm on it?
I'm just gonna hold this for a couple seconds. Okay, and I'll come a bit wider.
Let's see what we got here.
And then I'll get the feathers in there too.
Kill lasers.
Lasers off.
There you are. Cerametra, yeah, that's it. So Cerametra tricerialis is what we think the species is. It's in the family Xenometridae.
Okay, clear.
Nice sized boulder.
Can we pack that one up with a little ribbon around it?
Yeah, sure. Might have to put that into the open rock box there.
Yeah, that won't fit down here.
These all look loose. If any of these are of interest, Gad, lasers back on, Sean.
Yes.
What about that angular one up
Lasers
here,
are off.
Jasper? See this one at the top of the screen, Pilot? That's...
It looks an extra little bit black.
Well, it looks angular, like
Yeah.
squared off, rather
That
than
guy's
sediment.
That's a big guy.
Yeah, that one? Oh.
Yeah.
Where's the...
That
laser?
doesn't matter. I would just let Jasper carry it up anyway. No, that does look a wee bit big.
about this
I know. Okay.
How about...
Yeah, no, I thought there might be a chance, but maybe not. So a lot of this just could be broken off pieces of this manganese crust that's...
Yeah, that's when you see the slightly steeper sided hillside with slightly more angular looking blocks, that's better chance for a real rock.
So if you're just tuning in, we are diving on Alba Seamount, which is halfway between Guam and Wake. This is a live broadcast from the ROV Deep Discoverer, and it's being operated off the NOAA ship Okeanos Explorer. We are currently at just over 2,000 meters deep, 2,050 meters. And so we're pretty deep. We're 6,000 to 7,000 feet down. And we're looking at a very sort of mixed manganese crust. That's that black sort of rock material mixed in with sediment and boulders and cobbles and all types of things. And we're documenting the animals and the geology on the site.
Hey, Nav.
One of our objectives is that this is a site that's potentially going to be mined years in the future.
Yeah.
It's one of the sites that's been granted to the Russian Federation for exploration for manganese crust mining by the International Seabed Authority.
And
so we're doing a survey of the site, and I think this is probably the first time this has been examined and for the communities and for the rocks on this area. And, we're hoping that the data and video that we collect will be useful to both biologists and scientists and to the Seabed Authority and to the leaseholder.
.30 meters bearing 340 degrees at 0.2 knots.
You see one, Jasper? You saw a rock?
Yeah, I can't hear you, Jasper.
Nah, bridge move initiated.
Sorry.
It's okay.
Yeah, no, the one I was looking at looks a little bigger than I had thought it was.
Which one? Oh, okay.
See some more dead sponges. One's still attached and upright. The other is laying down flat right where the lasers went over there. Went over it.
Hey, Nav. Yeah, you're correct. Is that fix just closing and restarting the program?
Yep.
All right. Are you comfortable for us to do that now?
Yeah, good.
All right. Yeah. So just close the program and then just restart it. Yeah.
Mark that in the notebook.
Yeah.
Yeah,
I can get it.
You don't look that big until you see the lasers next to them. Then you realize they really are quite big. Oh, there's a nice coral. Big isolated
Pro
coral over there.
Do you like a view of it?
Yeah, sure.
Go,
if Jasper will allow a brief distraction from rock hunting.
Just try version 2.0.
I think there might be some coral people who are participating that would be happy to take a look at this.
Link partial.
Let me cycle sonar on vehicles.
So
is that safe to do?
Yeah, you didn't need that.
Tina believes this is a bamboo
Oh,
coral, family Isididae. Trying to see nodes on this thing. There were some closed up corals on the right too after we get some of the live ones, I mean open ones. There's one node there, so it's internodal branching. Yeah, you can see that. Yeah, so you will shortly, Tina, I think. You can see a node over on the left side of the screen, and there's a branch point right above it. And then you can see some of those closed polyps that were somewhere over on the right-hand side. Those are potentially informative.
Come on out a little bit, video.
Roger. Oh, I see.
Where were they?
There's some at the top and...
Oh, yeah,
on
there
that
they are.
branch.
Yeah, in the center, almost in the center there. Yeah,
just
keep zooming in.
Yep, come on in.
Yeah, that's...
And just to
and
the
slightly
left,
to the left there.
Branch in the
Pretty good.
background.
Okay, so what are these things called again? I should know what this is. Gotta look it up. They're called volcano polyps, if I remember Les Watling and Scott France's description of how this one group of corals retracts. What's that?
They all just contracted a little.
Oh, did they?
Yeah.
Okay, I think it's in the genus Ecnomyces where you get the contraction of the polyps like that, forming these sort of domes.
Copy that.
Yeah, I think that's a decent guess, Ecnomyces, for the moment.
When you're good, video.
All right. I'll get a quick establishing. Coming wide.
Okay.
Clear.
Full wide.
Nav bridge
Full
complete.
Thank you.
Yeah, you're at 8-0 now, pilot.
Some big dead sponges on the bottom.
So this area might be an okay area to go fishing for a rock.
Alright, is the ship moving right now?
It just stopped.
Okay. Let's hold off on another move for a second.
Copy that.
Shout out if you see anything.
Yeah, I think the stuff next to the boulder, the big boulder, is probably more promising.
Step on the that center up right now.
Yes.
Watch my back.
Wow, it's sort of like a little pedestal there. That's pretty interesting looking.
Yeah, it looks like whatever that was the top of drained away later.
Lots of dead sponges here for some reason.
Oh, looks either big or stuck. How about right next to that rock on the left and down a little?
Well, is that pretty big or?
So you're at
A little bit
9-0
further down.
now, pilot.
No, never mind, that's
Yeah,
stuck.
it's pretty. Yeah, it's pretty big too.
What was that, Sean?
You're at 9-0.
Yeah, I've got
I shouldn't swing much more forward though.
Okay.
Couple of sponges there. There's a Poliopogon sponge. We've documented that already. At least that genus, presence of that genus here. Yeah, nice colony growing right out of the side. Kind of a weird way it's coming out. It's coming out laterally.
It's like an old record player.
Oh, and there's a something that's similar to a Hyalus stylus. That particular stalked sponge there is pretty interesting. I think it's the first one of those we've seen.
On in video?
Yeah, and we collected one of these. I believe it was, could have been in the Marianas actually. It was either in Northwest Hawaiian Islands or the Marianas. Still need to figure out what it is.
Come in tight if you have it.
So see the osculum on top and then the stalk coming out the other side. So basically it's coming off, stalk's going right into the very base. And then there's a little hole on the top. Water again, even though this is a stalk sponge, presumably water is coming in the side and then going out that hole.
And Patty says that they got one on leg 3.
We have a touch
Okay, yeah, that's what I was remembering.
Oh,
These,
that's
so
it.
Shape looks almost like a tulip.
yeah. Kind of like a vase on a stalk.
There you go.
Okay, establishing. Okay, clear.
Right, coming up. Any of these?
There's any of those, Jasper? How about that one that lasers just went over?
Yeah,
the kind of square
slide
one.
to the right.
Yeah. Is that too big,
No,
Josh? Or?
no, that's good.
That one maybe?
Yeah.
Yep. Let's get hydraulic, Sean.
Can you do one of those brake jobs again, where it breaks
yeah,
in half?
absolutely. Yep. I can squeeze it.
Hydraulics up.
Well, that guy's cemented in.
I was talking about that one, yeah.
This one?
No, the one behind it. Or did you just push up
he
against
pushed it and it didn't move.
Sorry,
at all.
I thought you were pushing the other way.
Yeah. This smaller one moves a bit. What about that one?
That looks like a solid piece of manganese.
Mm.
Yeah, nothing else here?
No, I don't think so. Looks like he's standing on top of something else.
Yeah, he's a nice crinoid there. Look at how long the cirri on the bottom of them are. These are the cirri that he's propped up on. So this could be something like a telocrinus, because you see the very long tips of the arms that are barren, the pinnules on either side. That's one characteristic of a telocrinus. So I'm mark that down as a possible telocrinus that we documented here.
Okay, I'm going to come out for a wider shot. Clear.
There's a Phreid sponge. We saw a bunch of those already. Another stock crinoid. Yeah, a little of those, maybe those hyacrinus or something. So the train is flattened out, so it looks like we come up over the top of the steepest
we're coming
part.
up on top of the knob here, this little side part of the rift zone that we're on. It's gonna be ending up getting a little bit more and more flat, and then I think the end of the dive tentatively was planned where it starts getting a little steeper again. I don't know whether we'll get that far.
No, I don't think so.
Pilot, would you want to start moving again?
Yeah.
Or, okay. Nav, yeah, you can go 340. Keep doing what you were doing before.
Yeah, so how much more time is there left in the dive?
Yeah. I think, Bruce, we've got about an hour, maybe. I'm not really sure.
Okay, thanks.
Go ahead, Nav. $50 meters, 50 meters, bearing 340 degrees at, I'm sorry I didn't catch the speed, but I'm assuming 0.2.
2 knots.
Alright, understood.
Nav, what did you say was going to be the bottom time remaining? Thanks.
Nav, bridge move initiated.
Yeah, so I'll just relay that, Bruce, we have about an hour and a half, according to Nav.
Okay, thank you. That's helpful.
See a long-legged shrimp in there, and maybe a sea pen sticking up right by the lasers here. Might be worth a look, because we're getting into a little bit different type of substrate here, a little bit deeper sediment perhaps. And you can see the long legs on that shrimp there, and that's nematocarcinus. We basically see these things everywhere. It seems to be the most successful Certainly the most
Yeah, maybe
successful group of arthropods down in deep water.
Yeah, you can tell that we really have shifted from steeper terrain to the more flat terrain that really catches a lot more of the sediment.
One of the things I don't know is just how many species in that genus there are. You know, we just call all of these things Nematocarcinus, but we have no idea what, you know,
Taking a bit more zoom.
how many different types we're actually
in his face.
documenting.
Yeah, Patty's pointing out there's lots of tracks on here. This looks like there's been a lot of activity.
Yeah.
A lot of bioturbation.
Yeah, Tina suggests it could be urchin, or it could be sea cucumber, could be a variety of things that were
Taking
Sea pen.
Roger.
Come out a little bit.
so what is
Yep,
that?
come on in.
Well, whatever it is, it's overgrown. I don't think it's a sea pran. I think it's something that's overgrown with
Do you have any more?
zoanthids. But
That's full.
let's wait and see what others have to say about this.
Those spicules at the bottom, is that like a sponge stock maybe? I don't know.
It could be. Yeah, I see what you're saying, Josh. Yeah, could definitely be a sponge stock that's totally overgrown by these zoanthids. Yeah, and both Asako Matsumoto and Tina Molitsova both think that these are zoanthids. So, yep, could completely fool you. Yep, Tina also thinks it was a sponge as well.
Okay.
And Patty thought the trails looked like they could be sea cucumber trails, holothroidians. So,
They are funny
yeah.
shaped. They make zigzags here.
Right. A lot of dead sponge stalks that are overgrown by these zoanthids. There's a bunch of them here, 2 more that just swung into view.
Let's do a quick zoom. It's coming tight.
Yeah, same type of thing. So even when these stalks are dead, they certainly provide a function or provide a service to other animals down here.
Right, come on up. Just push ahead.
Forward.
Let's do a quick zoom here, just to snap. Alright. That's fine. Come out. Something in Sirius's view.
You're talking about the bottom left corner?
Yeah.
Yeah, it's gone.
It's a question for science. Depends on what they want to do. Point 3, you don't have a lot of time to zoom in on things.
Yes, you can ask wash leads if they want to cover ground or go a little slower and look at stuff. That'll decide.
Well, I mean, there's like slow look at stuff, fast look at stuff, and really fast, and do quick zooms on stuff. So we're doing the second one. Yeah, you can touch base with them. Yeah.
Would you go ahead, Nav? Yeah, why don't we do that? Because we're still seeing all, the same type of animals that we've already imaged before, so maybe covering ground would be a good idea.
Call Nav.
Go ahead, Neff.
There's a jellyfish.
Got a jellyfish.
I'm sorry,
coming
Neff.
at
Say
us.
again.
Come on in.
Yeah, I read 13.3.
Sure.
Yeah.
Go ahead, Neff. Copy, go ahead.
Kill the lasers.
I think this is a Hydro Medusa, but I'm waiting to see if anyone
All right,
else
I'm going to
wants
read
to
that
take
back
a
to you. I've got
stab.
50
Tina
meters
did.
bearing
This is
340.
hydrozoan
speed,
jellyfish.
decimal
Kill
three. Is that
lasers?
correct?
Yeah.
That's correct. Thank you, Bridge.
Copy that. Move is being implemented as we speak.
Lasers off.
Move is
For
initiated.
a bit more zoom.
So we've got Mike Vecchione who's in the chat room here. So he specializes
Copy that
in
now.
a lot of these pelagic animals. So be some good footage for him for his interests.
Okay, coming away.
Go. He is not cooperating.
There's a stock cranoid. That's a stock. Yeah, it could be, or it could be a different variety without those weird circular. So we're having a crew change here, a ROV watch change. So we'll be here for a minute or so while they get set up. And this could also be a sponge stock with just the unstock crinoid on it. Yeah, just hanging out. It's hard to say here. Yeah, yeah. Yeah, thanks, Josh. Got some big stuff in the background here. No, it's a big sponge or coral or something there.
Afternoon, watch leads.
Good afternoon.
Chris up here.
Good afternoon, pilot.
Josh passed on the word that you guys would like to take a look at this middle screen, or no?
Yeah, let's take a quick zoom at this and
Okay.
see if it's the same thing we collected.
Okay. Video, you can go ahead in. Try to center it up.
Yes, and
Okay,
it
holding
is.
partial.
It's the same type of crinoid that we collected with the circular discs going down it, and you see the little, I don't know
We
what
can
they
take
call
one
these
too.
little hairs that are coming off
Okay,
of
yeah,
the
go ahead.
arms. That's one of the differences between the ones that were the one that was collected in the Northwest Hawaiian Islands last year. It's a good shot of the actual center of the disc.
Interesting how uneven the arms and those side arms on the arms are.
Yeah, I think
It's
Okay,
very
gonna
long
come out for a
compared to
slightly
the total
wider
length
shot.
of that arm.
Okay.
Maybe it's partly human
Pilot,
angle.
I'm right above you now. I'm
Well,
not
no,
sure
I
if we're
think
still
also
moving.
that some of these got chewed off.
And
Actually, I'm ahead of you now. You seem to be moving
quick
fast.
establishing.
I wonder if he had that
Okay,
type
we
of.
gotta go here.
Yep, works clear.
Back 10 degrees.
Nav, what are we doing with the ship here?
Okay. I'm looking back
Okay.
max right
And
now.
what are we moving? 3-4, copy.
Yeah, that's where I'm pointed.
Okay.
You're trying to pick up a little speed over the flat terrain.
A little Period sponge, another one of these dead sponges with a lot of zoanthids growing all over it.
Yeah, should be.
Okay. I'll get out ahead here. It looks like Dave, you're looking back down at me.
Okay, that's straight down
nice
now.
little star-shaped imprint in the sand.
It's not really clear why so many of these are dead. But we saw this on a dive on the last leg, as I remember, where there was one place where we just saw a lot of dead stalks and dead stumps and things like that on one of these geos, yeah. Yeah, so Tina pointed out that even though it was the same one, the one we collected was in much better condition, which is really great. If we'd seen that one first, we probably would have grabbed it because it would have been unusual, but we saw the really good one first, thank goodness, and got that as our specimen.
Right, sure, out a fair bit ahead of me.
Yeah, another one of these. One of these. It's like Furia oca erectas.
Still have that tether cam flickering on D2.
Yeah. I'm not sure what's going on with that. Let's take a look at it tonight.
Yeah.
Tether looks good though. I can see
a couple live sponges in a mix with a dead stalk, overgrown. I think I got some type of a sea star on it.
Did you want to take a look at that?
Yeah, if we got a moment.
Yeah, we've got a moment here.
Sure, because I think this is different. I don't think we've seen this sea star so far today. We've seen the sponges.
Copy that. Come in for a quick look. Nope.
Oh, sorry. Misinterpreted.
Yeah, sorry. I'll help you with the video. Name. Okay, video, you can start to come in.
Roger.
Nav, has the ship stopped yet?
Yeah.
Okay, great.
Hold there.
Holding partial.
I'm not sure if Chris Ma is joining us right now. Chris Ma is an asteroid or sea star specialist at the Smithsonian Museum. But I'll give him a moment to take a stab at this. Otherwise, I'm going to throw out an idea that may be completely wrong, but I think is reasonable at least.
Is that a squat lobster underneath it too?
Yeah, and there's some type of a squat lobster. Yeah, right directly below it. Tiny little squat lobster. I think this is a... time's up, Chris. So the other Chris will throw out an ID. I think this is a type
Would
of
you like a
Henricia.
closer zoom, or is this?
The genus Henricia.
For now, we gotta go here soon.
Roger.
So his left bottom arm looks kind of lumpy there. Is that damage from running into someone else at some point?
Yeah, it could be. He could have lost it and then regenerated the arm.
Right.
You can go in.
You're right, because it looks...
You got
definitely looks kind of bumpy.
We could go up to the top, the left arm, right there.
I don't know if
next.
this is Henricia or not.
That's full zoom.
Maybe
I'll
Chris
just
will
do a
call
quick
in.
shot of the zoanthids in the
A
back.
little bit different
And the arms.
Copy.
Pattern. And these are zoanthids all over the dead stalk.
Okay, I'll max back at you now.
Copy. Alright, when you're clear video, we'll push ahead.
Roger. And good. Clear.
Nav, what's time off bottom?
So do the sea stars eat zoanthids as well as
yeah,
the
426
sponges and small?
UTC sounds right.
It's a good
I thought I
question,
had all that
Bruce.
set up right.
Yeah, I don't know
It's time
actually.
off
I think
bottom.
that's a really good question.
Is that right? Okay.
You know, next time Chris Maw calls in on this cruise, we need to ask him that.
Chris, in general, do you
Yeah,
like me
because
to
that stone
follow you
stock
left
looked
and
like it
right,
had
or
been
you
dead for
like
a
to just keep
while,
it
and there
down
wasn't
middle?
much else on there
No, you
for
can just keep it
the
down
sea
the
stars
middle, and
to
if you
feed on.
if you
So I
do
was just
follow
curious.
me, just let me know.
Yeah.
Yeah, it definitely could
Thanks.
be. There is a weird stock sponge too that was collected in the Marianas, I think. The weird vase, weird stocked vase.
Patty's pointing out more trails in the sand. There's a variety of different kinds, star-shaped and all over the place, wiggling.
At the end of that one? Yeah, I think so. I think that's okay with me.
That looks like
that's the idea.
Hyalonema sponge, which I think
Yeah,
it is.
that's
And
fine.
that would have
You're
been our
out,
first Hyalonema.
you're 45 ahead
Yeah,
now.
more stock crinoids.
Yeah, go ahead, Neff. We're out ahead a little bit. Do you want to take a look at this sponge here?
The one that was back a little ways?
Yeah, I'm just
yeah,
going to come
sure.
down right on it here. I think I'm okay with that.
Yeah.
Go ahead, RV. All right, let me copy this back to you. I've got 30 meters.
So we
bearing
collected a
335
similar looking
decimal
sponge
3
again
knots.
in
Is
the
that correct, or is
northwest
it 50
Hawaiian Islands
meters?
that had all these
Everything else is good.
zoanthids
50
growing on the
meters,
stalk.
bearing
And
335
I
decimal
think
3.
this
Stand by.
is Hyalonematid,
Holding
the
partial.
family Hyalonematidae. It tends to have a fibrous type of a stalk, but
Okay, you can keep coming in a little bit.
RV Nav, move has been initiated. 50 meters, bearing
the question is
335
whether it's the
decimal 3
same
knots.
thing, but I think it is.
Where's a good place to pick a zoom, Chris?
Well, this is a really nice shot right here. We can see everything we need, just with the whole colony. It's just that's just this cruise, and I don't have any stills handy to compare it to the one that was collected in Northwest Hawaiian Islands, but very similar though.
Finished with this shot if we'd like a close-up. Great. If not, we can. If not,
That's the
video's
first
clear.
of these sponges that we've seen on this particular dive.
I think
so
this is okay in video.
Roger. Pulling one.
and then we're still on a lookout for our second rock, right, Jasper?
Please pardon the interruption.
Yeah, but I'm not
This
sure
conference
that's,
contains less than 3 participants at this
that's
time.
going to happen until we
If you
get
would
to
like
a
to continue, press star
steeper
1
side.
now, or the conference will be terminated.
And we are moving at 0.3, so if you think you see one, let us know and we'll start acting accordingly.
So you'd be looking for some kind of big outcrop or boulder or something else.
Yeah, this is all pretty thin-skinned. It looks like. I'm guessing most of this is just manganese crust sticking out of the sand. Picking this up, I suspect we would mostly end up with manganese crust, not much else.
I'm gonna give you a little more tether.
Copy that.
It's kind of a larger boulder.
It's a larger
a
rock.
bunch of boulders right here.
Yeah.
You think any of these might work for a collection? 'Cause
Yeah, I've been fooled by the size of these things before.
I wonder if we could get the lasers on.
Yeah. Can we get the lasers?
They are on.
There they are. Okay.
Yeah, yes.
Right over there.
A little hard to see.
You see anything here, or you want them just to move on?
You want to go partial zoom video? Maybe we can just kind of scan around. Just hold there.
Sure.
I don't know if that helps watch lead or not, but
No, zooming out's probably better.
okay, come on. Try to work around to the other side of this.
There's a little coral right there on the side that's kind of interesting. It's that sort of pinky one that's right above the leggy one. Wonder if we could do a quick snap zoom on that.
Yeah,
It
sure.
looks a little different than what we've seen.
Okay, go ahead, video. Just do a quick snap zoom in. Which one of these?
Yeah, that's it.
Left or right?
Right center, right what you got right now.
I have a bit more zoom.
Okay.
Okay.
That's max.
Copy.
So I think this is a chrysogorgid, and we're looking at it sort of, it's a planar chrysogorgid, and we're looking at the end of it. So it could be Chrysogorgia crises, which is one of the planar ones that we know.
You just hold there. I'll try to get a shot so we're not right on the end of it.
Sure thing.
Okay, you can get back in.
Yeah, definitely looks like a Chrysogorgid. Just seeing if Masako or Tina are going to weigh in on this one.
Come on.
Going away. That's full wide.
Copy. Okay, you can come back in.
Yeah.
I'm starting to catch up with you. You're still fine.
Copy that.
Well, it's got some overgrowth there. Those are zoanthids that are growing on it. The actual polyps that belong to the colony are on the upper side, the upper part, as well as to the left.
Is that all you got, video?
Yeah.
Yep, that's max.
Copy.
So
There's enough footing there, sorry.
Yep, so Tina Molitsova agrees this is a Chrysogorgid, and it's got zoanthids on it.
Come out for the full organism.
Copy. Need to tilt up a little.
It's okay, this is fine. Alright, coming wide.
Copy.
Thank you.
Yeah, I was
Looks
just
like
suggest to the left.
Conolasmatine, I think.
Okay.
Okay, I still am.
Yeah, you're gonna swing for a bit. I'll push out. Center up. I think we got a corkscrew in it there.
Yeah. We sure do.
Are lasers still on?
Affirmative.
Oh yeah, sorry, just looking in the wrong place.
I guess the nice thing about dredging is you don't have to make any decisions, do you? You just get what you get.
Yeah, just deciding where to put the dredge in the water and how long to leave it on the ground. Yeah, I think if we want to get something here, we're going to have to go with a bigger piece in order not to have just end up with a piece of shell of manganese crust, essentially.
Kind of a weird little outcrop there.
Yeah, it's almost like a wave crashing over the side of this. But lava usually goes the other way, so it's probably not a wave.
It's like we're getting into a little more, steeper terrain now.
It's starting to get more rocky again.
So you had sort of a little flat patch there, and now we're.
Is
that what it says?
Probably keep us moving now.
Yeah, the next contour closes and then
I've about stopped.
Yeah,
we
do
did the
same
flattest
thing,
part of it.
We'll just wait to hear. I'll stay ahead a little bit maybe, and if they want to get a rock, we'll just pump the brakes.
Go ahead, RV. All right, that's a good copy. I've got 50 meters bearing 335.3 knots. Is that correct?
Good copy.
All right, stand by.
These are pretty large pieces of rock, if we would ever be able to get them loose.
Some of these look like they're pillows. They sort of do.
Yeah, they have. Well, they're tubes more than they are pillows, I think.
Oh, I see. Okay.
These things form underwater. Sometimes they just make a little pillow lava, or sometimes they keep flowing and pushing out and make more of a tube-like.
RV Nav Bridge, move has been initiated. 50 meters bearing 335.3 knots.
I'm gonna have to wait a minute. Could maybe try to snap on this fish though.
Ah, there's a fish. There we are.
Way out at the end of my tether, but looks like it's coming at us. Go ahead, video.
Zooming.
Still 20 meters off the
Hold
line.
there.
It's a different synaptobranchid, so hopefully Bruce will get a look at this. I think it's different. He might think it's the same.
Can we turn off the lasers?
Well, the dorsal is kind of far back, but.
This looks like another synaptobranch, but it does look a little different from the others that you saw earlier, so we may be looking at a different species. It's going to be hard to get a good close-up of this, but again, if you can get a close-up of the head and the side of the body behind the head, that would be really useful. It could be, now that I see it from a different angle, it could be the same species that we've seen before, whether it's Synaptobranchus dorsalis or Synaptobranchus oregoni is something we need to try to determine. This is certainly more active than some of the others that we've seen. Behaving a little bit differently, but that may not mean anything for the species identification.
Very curious.
Again, it's got a sort of a little damage to its snout, a little scarring on the body, like we've seen before. But it's a cutthroat eel in the genus Synaptobranchus.
Yeah, thanks, Bruce. And that was Bruce Mundy, who's a deepwater fish as well as shallow water fish expert, who's based in NOAA Fisheries and the Noye Regional Center on Ford Island. He's operating the IRC Exploration Command Center over there.
Patty Fryer made a comment on the chat room. The behavior that we saw a little earlier, where the thing kind of stopped and flexed its body, and I think maybe opened its mouth. She was asking if it's a threat gesture. And we see that a lot with these eels. As the ROV gets close to them, they seem to have a response that very well could be an aggressive type of reaction, or they may be flexing their body, opening their mouth to lunge and bite at something that might be a predator that would attack them. They're, again, very active eels, and they're lunging predators, so they also may have that sort of defensive reaction. They feel threatened. It's an interesting kind of stereotypical behavior that we see in these cutthroat eels.
On the west side.
Yeah, I always thought it was something that to do with them being bothered by the lights from the ROV, but maybe not. Maybe it is some type of a behavioral gesture.
And it could be something that the lights elicit, that normally would be elicited by just the approach of something large, but there could be any number of stimuli that would produce that reaction on their part.
Come on, Jasper, there's got to be a rock for you here. There's got to be. Speak up. There's rocks all over the place.
There's rocks all over the place.
It's Rock City.
We just want to make sure we pick one that's actually a rock.
That's true, rather than a crust.
Yeah, and then the problem seems to be that anything that I would say, yeah, that's probably a rock, ends up being a half meter across.
Operations being held on the bridge in 5 minutes. Say again, oxygen on the bridge in 5 minutes.
Yeah, what was that last move? 340? Oh. Okay. We should probably next move go more north. What do you think, Dave?
Yeah, it's maybe 10 or 15 degrees to starboard, looks like.
I don't know. What do you think, Jess? Are you seeing? You can see the lasers anchor out a little ways.
Lasers are off.
See where they are.
Lasers
There they are.
back on?
Okay.
There they are. Yeah, see, that's what I mean, is the rocks end up being a lot bigger than you think they are.
Yeah.
Yeah, maybe the round one to the right and up a little.
It's pretty big.
Too far to the right.
He's talking about that minivan size rock. No.
Which, I think directly to the left of your lasers.
He's talking about the one to the left of the lasers right there. That looks, right there.
Yeah, that's the one.
It looks, it's a little big, isn't it? Or
I think it's a little big.
it's kind of hard to get at it too, isn't it?
Yes. Yeah. Depending on.
Yeah, what's the long direction?
Yeah, that's I think that's a little big. That's probably not all of it anyway.
No, we'll look a little further up maybe.
Okay. Okay. I think we could do another one.
Sure.
Yep.
Surprised we're not seeing more animals growing on these rocks. It looks like they'd be fairly suitable. They're large enough to be pretty stable. Get an occasional crinoid like down there. There could be a little shrimp or fish in the water column straight ahead.
Be further to
A
starboard.
white thing. Maybe one of these.
Maybe due north.
Maybe a shrimp, perhaps? I don't know.
Hey, you want to start to come in video? I think there's two of them there, just kind of very, very small.
Sounds good.
Yeah.
I'll try to get them in the black background here. Looks like a little shrimp.
Yeah, waypoints due north, right?
Oh yeah, a little shrimp. Okay.
Is that enough for an ID there, Chris?
It's enough of an ID for me since I don't know him.
Do you have any more video?
Yep, I'm about halfway in.
Oh, okay. You can keep coming.
Okay.
If you can track that.
I think he's one of these. See, this is where Scott France is needed. He knows what all these weird little creatures are that are
come back to him.
Swimming above the bottom.
Come wide. We can. I think we can let that guy go.
Sounds good.
Yeah, I'm good with that.
I said, yeah, there we go. Mike Vecchione's on.
Arby.
I forgot. Thank you. Yeah, thanks, Mike and Patty.
All right, copy that. I've got 50 meters bearing 000.3. Stand by.
So Tina spotted another one of those stock crinoids with the round discs on there. So there seem to be a fair number of them here where they, I mean, we saw them that one time last year, and I think it might have been in Johnson Atoll, and that was it. We'd never seen them. We'd never seen them before, and we never saw them after that one time. Oh,
very bright.
But
RV
they
down
seem to
bridge.
be
Move has been
reasonably common
initiated.
here.
50 meters bearing 000.3.
Looks like a sponge or something.
Can't tell.
Whoa, look at that guy.
Anytime I see it first in Sirius, we know it's going to be big.
Yeah.
And could we look at the coral before we get to the sponge, this little branched one? I think it's a black coral. It would be the first one that we've seen on the dive.
This move? Is that interesting enough to...
Yeah, probably. We're going to probably take a while at the sponge, too.
That was my guess, yeah.
Yeah, you can go ahead and stop it.
Go ahead, RV.
Okay.
Yeah.
I copy that. I'm going to bring the
So
ship to a
Tina
stop.
Molotova has
video, go
identified
ahead.
this coral as
Okay.
a Bathopathes.
That's a partial.
Copy.
Nav, how far did we move before we canceled that?
Keep coming whenever you're clear.
So that's
I was
full
wondering
zoom.
when we were
Copy
going to
that.
see some black corals on this dive. I was worried we wouldn't see any.
Oh,
That's
okay, just a few meters. Where you might
finally
not
got
have
one.
Tether. I think I'm still swinging from the last one, though.
beautiful image of one of these things. So they basically are, these are a hexacoral, not an octocoral. They have 6 tentacles, not 8 tentacles. And 2 of their 6 tentacles are exaggerated in length and size compared to the other 4. And that's what you're seeing on either side. It looks like you're seeing little branchlets coming off of these branches, but they're actually the tentacles of these polyps, the 2 enlarged tentacles. And the polyps are kind of lined up in a row right along the branches there. So they have a very distinctive look, and there's something right in the middle. Is that a worm or something? That's right.
Darker
Yeah, with the
spot
polychaete. Okay. Yeah, Tina had already spotted that.
Can you give me a little tilt down, Dave?
Oh, on yours?
Yeah, on mine.
Sorry.
Thanks.
Sorry.
That's all right.
I zoomed in your...
That's good right there. Did you want a little closer shot, Chris? That's full zoom. I can move the ROV.
No, I think that's adequate right there. That would be fine. We don't.
Remember, he had a sponge lined up next.
Yeah, we have a sponge, and then I guess for some reason we need to grab another rock at some point
Yeah,
too.
you-
So
you need at least 2.
Okay. You want to come wide when you're clear, video?
All right, coming wide.
You want your camera back?
Yes, please.
All right, full wide.
You have it.
this is definitely in the genus Polyopogon in the family Pheronematidae, a big, huge glass sponge, a beautiful specimen. If you look at the way it's attached to the rock, it's got a fibrous attachment, kind of anchors itself with these fibers. And that actually eliminates a lot of different sponges as candidates for this one, plus the size of it. There are not that many different types of glass sponges that get this massive. And members of the family Farinimatidae do get this big. So we can pretty quickly narrow down what type of sponge this is. Now, I don't know what happened to the end of him and why he is so weirdly shaped.
He's just bent over. He's old. He needs a cane.
I think that's it. He sort of looks like I feel sometimes getting out of bed in the morning, that's for sure. He's oftentimes, even though these sponges are massive, they have a little more symmetry to their body forms, but this guy is really, you know, he's got a very unusual shape to him.
Bruce was asking what that was.
That's a crinoid, I think, on the back side of him. Could be a little different crinoid than we've seen so far. And then you can clearly see how fibrous the base of it is. And so that's just a dead giveaway. And there's well, most of the sponges that have that type of attachment are in the a particular order that's called Amphidiscophora. So,
anything in particular here you want to take a zoom on?
I think we're good. I think we're good just with this. Thank you.
Sure. Let's get that shit moved back in.
Go ahead, RV. That's a good copy. I've got 50 meters bearing 000 decimal 3. Stand by.
So I'm just taking notes on the different types of animals we've seen on this dive. This is for the dive summary report that we need to write this evening. And I'm already on 2 pages of animal notes already. You know, even though they seem to be pretty sparse and spread out, we've had a pretty amazing diversity of different things that we've seen today here. So it's a lot of different types of sponges. We've got a variety of unusual crinoids, and we've got a bunch of different corals, bamboo corals, the chrysogorgid corals, some anemones, big, huge, whopping anemones. One of them which had us completely stumped.
Quick heads up. So the last time we were running at 0.3 knots, and you said, you know, go ahead and stop here. That puts an enormous strain in the engines to go from 0.3 to 0. There's really no way to kind of step it down. It just goes 0.3, and we're telling the ship to stop. So if we could try to mitigate that or cut down to like 0.2 or things like that, it might make life a little easier.
There's also something red to the right after this.
All right, thanks. Bye.
It might just be in the rock face. I was just checking. While we have some time. Okay, come wide. What something
there
red?
There is something red. There it is. And I think it's an Anthomassus. That would be the first of those. In the upper right.
Yeah, I see that, yeah.
And we happen to have participating today one of the world's experts on these particular types of corals.
Can I go lasers on as you approach this?
Yeah, sure.
And that is Tina Molitsova, so I think she could enjoy getting a zoom on this and be able to help with the identification.
Okay, video. You want to start to come in? Hold there.
Yeah, first one of these.
So that's
Good.
partial.
Copy that. You take my camera, dude.
Want me to follow it or just stand by?
Just stand by. Just give it one little tilt up. There. And that's a partial video?
Yeah.
Okay, you can come in full.
Alright. So that's full.
Copy.
He's got a little friend right next to him.
We are waiting for Tina to weigh in on this animal. Probably has a little bit of a delay on her video.
Looks like there's a shrimp or something.
Patty Fryer, while we're waiting, just noted that it's a really gorgeous animal, and I agree.
Yeah.
These are just beautiful. So they're called mushroom corals, I think.
What is that?
It's an isopod, maybe?
Yeah, isopods. So Tina, you want to weigh in on whether this is Pseudanthemastus or Anthomastus?
I'm oscillating on my toe, sorry. Give it a little tilt down, Dave. There you go, hold there.
It's hard to tell from this angle, I think. Sometimes you need to see the
the polyps inside of them?
The stalk is the...
Oh, okay.
Yeah, so Tina says it's in the genus Anthomastus, but she can't go further than that. Yeah, thanks, Tina.
Okay, come on, video.
Okay, we're wide.
Bring your camera back. Copy that. You can maybe do one quick look at the polyps here.
I'm catching up.
Yeah, and then we gotta go.
Gonna give you a side view here, Tina. See if that'll help with your ID.
Okay, video, come on in. Hold there.
Alright, take it again.
No, I got it. Keep coming.
Alright, that's full.
Copy. Hopefully that can help a little bit.
So this is a beautiful image of some of the characteristics of octocorals, which this is. If you count the tentacles on each of these very elongate polyps, you can see there's 8 tentacles. And these tentacles are pinnate, which means they have little pinnules coming off them, the little spikes coming off of them. So when you see this type of arrangement, you can be pretty comfortable that it's an octocoral. Those of you who've attended other dives have heard this story before by numerous participants. But for some of the new people there, this is just one of the key characteristics, the first thing that Cnidarian experts look at.
Okay,
So
we got
try to count the polyps
another
and see
couple
whether or not
seconds
the tentacles, and try to see whether or not they're pinnate or not.
Come wide, video.
Alright, we're full wide.
Copy that, thank you.
Okay.
Okay.
Another one of these same big polyopogon sponges that we just imaged a moment ago. All right, what about what about these rocks? These might gotta be something there. Looks like a Candidella gigania, that unbranched coral that's in the middle of the screen. We imaged what we thought was that species earlier.
Something in the water column, or
it almost looks like it's on a stalk.
Yeah, it is. Yeah.
Yeah.
Check it out. No rocks here, Jasper.
I'm max back at you now.
Copy.
Okay.
Is that one too big, Jasper? This one here, or? Oh. Well, what about that? See the crinoid, the yellow crinoid, and if you go just slightly to the right of that and up slightly, there's kind of an angular rock. Would that be too big?
Would you turn to follow? Turn to give me a view, Dave.
Follow you.
It's directly above the lasers, a little ways above the lasers. That is.
That one there, or the one right
keep
to the right
going and to the right just a bit, a little bit further. You're almost on it.
There.
Yeah, next one up, because that one's a monster.
I think you're muted, Jasper.
Is that too big right there? The one the laser's on now. It's pretty big.
That's about 30 centimeters. Yeah, a little bit about this big.
Well, I don't know.
I'm having trouble finding you.
You don't know the depth, but it's about this big.
Yeah, there you are.
Yeah.
You're due east.
Pilot, do you think that's even free or not?
Let's take a little closer look here. I didn't want to hit that stalked creature. Can you do a partial here video? And hold there.
I don't know.
Looks large. I don't know. It's hard to tell.
No, I don't know about that.
It's a pretty big one.
Seems pretty large.
I
Wide video.
think that one's going to put up a fight.
Yeah, me too. Come on,
Nice
about
holothroidian.
A little bit to the left, maybe.
A bit to the left.
And a little up.
And up.
Kind of hard to get at, though, up
Yeah.
in there.
Which, we can get at those. Which,
What about further to the left over here, right on the other side of this little formation coming out? What about one of these?
Those are going to be similar in size again.
Are they?
Yeah.
What about, like, that one down there? The one underneath this one that the laser's on? Is that not enough of a rock?
The one that the laser was on, or the one under it?
Let me get back
There's
to
an angular one here, directly under the laser.
That
That
one.
one, yeah. Would that work? Or not?
Can you squeeze it?
Yeah, we can. You want to go ahead and do that?
Yeah, the slightly one to the right one. See if we can squeeze it in half.
Okay. Sure.
If not, it's probably good. If so, then probably not good.
If so, we won't be collecting it anymore. Okay. Let's get set up here. You want to bring up hydraulics for me?
Yes.
You think we're stable?
Yeah, I think we're okay.
Okay.
All right, there's 1500.
Copy.
Video, can you give me a partial zoom on Sirius HD and get a
Yep.
fairly rare view here? That's good.
How's that?
That's perfect, thank you.
No problem.
tether's trying to spin me. That's why. That's alright. You can't really do too much about that.
Here. I can give you
Yeah,
a
I
little
was
bit
just
more.
looking at that overhead view there. Those of you who are, who've got the 3 camera views, if you want to, oh, there's a fish right there. It's going right in front of the. It's high up. Yeah, Sirius picked it up. Yeah, it looks like it's another synaptobranchid. We were pointing out the camera 2 feed from the Sirius looking down at the ROV at a pretty interesting view of it.
Alright, can you bring up, I don't know what would help. Maybe Titan? Maybe Mini-Zeus?
My screen?
Yeah, please.
Why don't we try to type?
Alex coming live.
You want me to do anything with it?
Yeah, bring it over to center screen maybe.
It's full zone.
Copy.
Full wide.
So if you're just joining us, we're about to squeeze a rock. This is what we refer to as the contour test.
Yeah.
It's a reference to Jasper Contour, who's sitting on my right, the geologist, the onboard geologist for this particular cruise.
Force 8. That sounds good.
And he's
He usually doesn't get to pick his rocks this way. He usually gets a dredge basket to fill.
Okay, it is fighting me, and not anymore.
And it's not a rock.
It did not pass the test. And the consequences were severe.
That's right.
You follow me home there.
Yes. You're not going to try anything else here?
I don't know, any other candidates out there?
I don't know. You want to just keep moving on and try to find a little better area, or you think there's one of the other ones might?
I'm trying to see how close you are, like how much closer you can reach.
Oh, I mean, I can bump in. That was easy to reach.
Okay.
If there's one close.
So we have about 25 minutes on the dive, left in the dive before we're leaving bottom. And I am just noting this because in the chat there was a little question about that.
Jasper, anything else here?
They're either huge or tiny, and the tiny ones probably won't have much in them.
Well, we're set up if you want to give it another go on one. I can nudge him.
We have a very unique Sirius view of sampling here.
Yeah.
I'm looking.
You can switch back to that if you want. While we're looking.
Hey, Emily. If they start to sample in the same place again, why don't you leave it on
you look up a little
SLED
bit
HD? Just because this is, you don't usually get this kind of view for that.
Sort of triangular shaped. Keep going a little bit.
Well, I'm sorry. Go down a little bit.
No. You were going right.
Oh yeah, I think I see which one you're saying.
Most
like
left.
directly left the lasers now.
Yeah.
Might be a little large, but
it's a pretty cool perspective. Just looking at the rocks.
Certainly find out.
Yeah, it's reasonable size.
Yeah, it might be doable. It'll be a little tricky.
Oh, it's got a
grab.
depth to it.
Oh, does it?
One of these big round ones would be nice.
I think it's well, you can look in the port cam there. Yeah. Yeah, it's way below me. It's hard to find a good spot to grab this guy. We can just hover there too.
Yeah, so
the one dead center here. Yeah.
Bruce had pointed out again the Sirius camera view. For those of you who are interested in switching to camera 2 from camera 1, you get a kind of really nice view of the ROV position in front of these rock formations that we're
If you want to just keep an eye
looking
on it
at.
let me know.
And Tina had mentioned there's a shrimp, and yeah, we saw a shrimp just a moment ago.
Gonna lock on some rotate here, putting the tether.
And the tether still looks okay.
Yeah, it's just trying to swing me.
Oh, right.
She says there's polychaete tubes. Maybe these two little things sticking up in the sediment is what she's referring to. You are, if you just joined us, you're also listening to a live broadcast from the NOAA ship Okeanos Explorer. We are diving on Alba Seamount, which is about halfway between Guam and Wake in the middle of the western Pacific Ocean. And we are currently at a depth of about 1,995 meters. We're getting toward the end of the dive. We've collected 2 biological specimens, and we got 1 rock sample, and we're trying to get our second rock sample. And while we haven't seen a huge abundance of animals, we've certainly seen quite a diversity of animals here at this site. So it's been a pretty interesting dive.
Okay.
Some things are familiar to us, and a bunch of things that we hadn't seen before.
I might need that. How do we fix that?
I don't know how to fix that. I can put it up here.
Okay. I mean, I won't need it up there, but.
Okay. Well, should I put pilot up on my screen?
Yeah, I think that'd be good.
Let's see. Follow you out? Or do you just want it centered?
Yeah, you can follow me out. That'd be good. I don't think that this guy will crush. Do you, Jasper, or is that like knock on wood?
I think looking from the top view, it looks like it probably won't even come loose.
Okay. If that's the case, then we should open the port box first. There is something in the starboard, correct?
Yes, there is.
Come here.
Oh, you moved it.
This is the contour test. Okay, I don't want to.
Feel it.
This will be our rock.
I think that's good. Fortunately, we need to open that box.
Yeah, it's okay. There's the rest of it.
Yeah, it looks pretty crusty.
Okay. Is that good or bad?
Can you turn it out of the cloud, the bottom and forward? No, you can't do that too much.
We can try to. We can do that. Does that help?
Yeah, the other side of it on the bottom. Yeah, there we go. That's all crust. Yeah, I think so.
So
I think
do you
it's all
still want
crust.
it? Do you still want it, Jasper?
It's all crust. I'm not going to do anything with that, but other people might. Yeah. So.
So, collecting?
Yeah, I think we'll just collect it because I have a feeling we're not going to get to collect anything else.
Okay. Can you go to the port rock box here on the Titan? Sorry.
Oh, yes.
I just want to see if I can lift it with the rock. I'd rather not put it back down, but if we have to, that's fine. Keep going up. Okay. I don't want to.
So while we're putting this rock away here, I'll just update you on what's going on here. We're on the NOAA ship Okeanos Explorer. This is a ship that's operated by NOAA's Office of Ocean Exploration and Research. It's a NOAA office. It's designed to go to places where no one else has gone before and find new things in addition to doing some really good science. The ship itself is operated by NOAA's Office of Marine and Aviation Operations. And the ROV operations are, I didn't really say that right, but for ROV operations, our partners are the engineers from the Global Foundation for Ocean Exploration. And it's this superb group that operate and maintain the vehicles that we're using today, which, by the way, in my experience, are the D2 and the Sirius are the most stable and marvelous ROVs that I've ever worked with. Just when they go in the water, you can depend on them. They get beautiful imagery and do a tremendous job down here all the way to a depth of 6,000 meters. So the 3-year program that we're in the middle of right now is called Capstone. It's a program designed to actually provide some exploration and science for the U.S. Pacific monuments. Right now we're on Alba Seamount, that's between Guam and Wake. We're coming from the Mariana Trench Monument and we're headed toward the Wake Monument, and along the way we're taking some time to dive on some sites in international waters. These are exploratory mining sites. And this particular site that we're on right now has been granted to the Russian Federation by the International Seabed Authority to do some exploration for manganese crust, which you're seeing a lot of right now. And then tomorrow and the next, let's say, I think we got a transit day tomorrow, but then the day after that, we're going to be diving on a lease site for the a Japanese group, and it's for the same purpose, for exploring manganese crusts. And we'll do 2 of the Japanese sites in collaboration with one of the people who are in the chat room right now, Kazuya Naito. Mr. Naito is representing the Japanese interest for explorations on that site, and we appreciate him participating. And then once we complete the 2 sites on the Japanese lease blocks, then we are going to be heading into the Wake Monument. We'll be doing a series of about 11 dives in the Wake Monument before heading back to Kwajalein, where this cruise will end.
That's full in.
Copy. Yeah. It is. I'd help if my arm wasn't in the way.
So Shirley just read this, didn't she? I can't possibly remember all this stuff. She had to have read. Okay.
Jeez, it seems like one of my joints is happy here. Hold on. Yeah, maybe.
I know I'm live on the mic here, but I was just commenting on this because I'm trying to follow Shirley's amazing sort of partnership spiels, and I can't do it. You know, trying to read all of this. She did a much better job of actually providing this, so I'll need a little practice before I get this down a bit. Yeah, that's the point. As
okay, I'm moving out.
Roland here sitting next to me is pointing out, the main message that I was trying to convey here is the fact that for an expedition like this, there's an awful lot of people involved and different organizations and entities. We have scientists on shore participating, various groups here on the ship as well that are responsible for different parts of the operation. So it's pretty complicated. And I just butchered the giving credit to all of these groups, but I will do a better job on the next few dives until I get this down.
All right, I'm going to take my monitor back.
Copy that.
Video, can we go full wide on Sirius HD?
No problem.
Thank you.
There you go. Full wide.
And we are fairly twisted up, as you know.
So this is Bruce again. While we were picking up the rock on a couple of the screens, I could see another one of the cutthroat eels swimming around. It was another Synapple brancus. And at this point in the dive, I would say if we see more of those, we probably shouldn't zoom in on them because it would be better to cover some more ground to see if we can find some more diversity in things.
Sure, thanks, Bruce. Yeah, it'd be nice to see a macruid. We have yet to see a single macruid. We've seen one cusk eel here, no rat tails, and we've seen these eels, and I think that's about it for fish, if I remember correctly.
East and actually the south.
Okay. I know when you end up
Yes,
around
that's
15
right.
or
And
so,
that's actually one of the remarkable things about the dives in the Marianas and here compared to the Hawaiian archipelago is the how scarce these rat tails or grenadiers, the macruids, are. Because usually they're one of the more abundant types of fish that we see at these depths.
Your auto heading is in.
Pass this real quick.
And did you get all that recorded before you switched?
Yes.
Great.
Yeah.
Okay, that's a bit better. Come back to you, and then
Okay, you're more or less directly below me, so if you head out at this heading, that should put us in good shape.
Okay. Let me go around this guy.
Once you pass me, I'll turn to port. That'll take out a third of a wrap or so.
Okay.
So in the meantime, we've moved on from our collection site. We picked up a large piece of material, likely mostly manganese crust. Did not look like there was a lot of volcanic rock attached to it, but we collected it anyway. There are other folks that are interested particularly in the manganese crust, so we have collected it anyway.
That's good.
Okay.
And we are headed out.
I'll keep on heading forward.
Our last few things before we are done with this dive.
And we're going for 1.5.
Yeah,
ultimately.
roughly around there.
Great.
We'll take a look at it and see. Okay, I'm going to follow with you now. Well, it kind of looks like it goes down slope there, honestly. Yeah. Hey, Nav.
Your mic's a little low.
What's that?
That's better?
Yeah, that's better.
It's looking downslope that way, so I'm thinking, should I stay on top of the high right here?
Yeah, probably.
Okay.
Yeah, come a little more east.
Okay. Then cancel my last thing about 1.5.
Okay. I'm going to follow you east.
Just let me know.
I'm going to go ahead and get up here a little bit. We're a little heavier now. I'm going to reduce our Z bias.
Yeah.
Okay, so a little update on the post-dive planning call. So we may just say a few words. It's going to be very brief because we do not have very good multi-beam data on our next site, which is not going to be tomorrow. It's going to be the day after tomorrow.
Give him a little look.
Take a
And
Little
maybe
peek
we'll
here.
take a look at this purple sea cucumber first, and then I'll finish what I was saying.
I'm going to do a quick zoom video.
All right. That's partial in.
Thank you. I don't know if we need a full zoom or anything, but if you want one, we can.
It's up to you.
A watch lead.
Roger, pilot, go ahead.
I didn't know if you wanted to full zoom on this or if you want me to just continue on.
Let's go ahead and continue on.
Okay.
Thank you.
We'll come on out video whenever you're ready.
All right. Full wide.
All right.
So the not tomorrow, but the day after tomorrow, we're going to be diving on I think it's an unnamed seamount.
Up a little higher.
It's in the area where
Actually,
there's
it's
okay.
some
go
ISA
forward.
exploratory lease blocks
moving animals.
that were provided to the trying to remember the company now, to tell you the truth. In Japan, it's a Japanese ISA lease block.
Got some
But I think
sponges and
there's
whatnot up here too.
I apologize. I've got to look that up. But in any case,
appear to be
we
back
don't
on
have
the-
very good multi-beam
the high
data
here.
on that site.
Yeah,
There's some 1980s
Yeah.
multi-beam
25.
data that
Yep.
we found,
Well in there.
but
So
it's not
waypoints.
that good to actually
north.
select the dive site. So we were
But,
going to try to
at
get
this
up
point,
there
I'll
and do
keep
some
Yeah.
mapping ahead of time before
Okay,
we
I mean,
pick
if you
the
guys
dive
want to do a
site.
ship move roughly here.
And,
We've only got a couple minutes here.
Okay, I'll just fly around.
Yeah.
Okay.
Then
How
it'll
many
be
minutes, Chris?
pretty sort of
when we
a
have to
rush
leave
dive
about
planning call once we
10
have some
minutes
data
from now.
to figure out exactly a good
And that
site.
factors in a little moving around.
Yeah.
Okay, I'll
Great.
go explore
Yep.
then.
Yeah, go find something cool here.
And then, of course, the issue with the 1980s
little
data was
tether.
that it's also very limited, not
thank you.
only in its
Yeah, it looks good.
technology, but mostly also in how much it actually even covered of the seamount that we're looking at. Chris, is that right?
That's correct.
Yeah. So.
Any of this have any interest to you, watch lead?
No, this is all pretty dead.
Okay.
A variety of dead sponges.
You got something in the water column up there.
Right there, that pink.
Yeah. Get that guy.
Do a quick zoom.
That's a partial zoom.
Okay.
I guess it's some type of hydromedusa, the hydrozoan, it looks like it.
Oh.
Being pretty lazy, he's certainly not pumping his bell very well, is
Nice,
he?
Chase. He's running.
Change elevation.
Ah,
Come down on it.
yeah, should have done that. Okay, go ahead and zoom out.
Right, full wide.
Okay.
That's all good. You got a good shot.
Okay. So now I'm going to give the proper entity here, and so this is Kazuya Naito, who's on the chat room right now. So Mr. Naito, I apologize for not remembering the exact division you're from, but he is with the Seafloor Mineral Resources Survey Division of the Metals and Mining Technology Department, the
I'm getting
Japan
kinda far
Oil,
out there.
Gas, and
Yeah,
Metals
that's about all
National
you got.
Corporation.
Okay.
And I've pretty
So
well built
this
already.
is the corporation that has the leases in these areas where we're going to be doing the next couple of dives.
Just
So
more
I
dead
will make
sponges
sure
there,
that I
looks
remember
like.
that when we get there. And so we are absolutely delighted that Naito-san is participating on the dive today, and will be participating on on the next couple of dives as well. And so the intent here is to actually provide very, very useful data to all types of entities, to this particular corporation, to the ISA, and to other biologists and geologists who are interested in these Pacific flat-top seamounts or geodes. So the more uses we can get out of our surveys, the better.
I was gonna say, yeah, maybe lateral that way, and then see if you can see something on the other end of that, on the other side of that little cliff. Once you get a little closer to Sirius.
Okay.
Watch lead nav.
You want to follow me down? Yeah, sorry.
Roger, Nav, go ahead.
Hey, Chris, 5-minute warning.
Roger, I got that. So we got 5 more minutes. So this is when the giant squid is going to show up, correct?
That's what we plan. That's the plan.
I'm following you to the east. Okay, I'm just going to... What do we got here?
Heather still looks good.
Want to give me a quick zoom?
Those look a little bit like pillow formations there.
You want to zoom?
Yeah, just
yeah,
there you go.
This looks very much like just thickly covered pillow and tube-like structures. It's just so thickly covered we can't just take one out.
It's tricky. Okay. I'll zoom out.
All right, full zoom.
Thank you. Let's go take a peek over here.
We're seeing some dead sponges. So when we see these sort of big dead sponge skeletons, and it's quite possible that they either just got too top heavy and fell over and eventually died, or maybe they just got too old. See a fair number of these particular sponges that I think are in the genus Aspidoscopula, this dead one that's right in front of us here now, and I'm sort of viewing this as sort of a sail. So if there's a strong enough current coming along, that's going to provide a lot of resistance. It's an upright sponge with a lot of leaves coming off of it, and maybe that's the problem. It can get up to only a certain size, and then it just gets knocked over.
Is that this one that he's talking about?
This also looks very narrow, actually, of a ridge.
I'm not sure.
This is.
I think it is.
Okay.
I can't tell exactly, but you can sort of see the other side on the left already dropping down again. So this is a pretty sharp sort of ridge that we're looking at. So that might expose it even more if you want to think of it that way.
Some of these corals, they look like some of the same primnoid octocorals that we've seen previously, probably in the genus Narella.
Any suggestions on where to fly?
Well, there's a sea star there on the bottom. I wonder,
probably
just to the left
star.
of the lasers, yeah. One last zoom.
Where is this guy? Left of the lasers.
Just left and above.
Left and above? Oh, okay, I see him. Yeah, let's go ahead and zoom in on that guy.
All right, just let me know when you're ready.
I'm ready.
Okay. You want a full zoom?
Sure. Try to keep it steady here.
Yeah, you get one more last good shot here.
Yeah, I don't know what this guy is. He's a different one.
Shot
Yeah, he's got a little tube feet sticking out the end there.
Get Chris Mah on the horn.
It looks like he's stuck to the wall there with him.
Yeah, so he gets around. So after the ROV lifts off, we're going to have a very brief, I don't think I actually completed this statement I was making earlier, but we're going to have a very brief post-dive call just because we don't really have any good data to show you where we're going to dive the day after tomorrow.
Okay, come on out.
So those of you who'd like to stay on the line for a moment, we'll just do a quick wrap-up.
Yeah, you want to
brief
switch places?
Update on our plans for the next couple days.
Okay.
And then we'll
I'm aligned with the ship. You
go ahead and sign off.
Turn.
Yeah, we probably want to come up a little bit first.
Sure.
Hopefully you'll rejoin us in 2 days from now.
Oh, okay. I just couldn't see it from where I was. Okay.
I have a half negative thing.
Good.
Can you scroll? Can you go down just a little bit? Keep me in view. I can also push out a little bit, but
no, that's fine. Ready for me to start to come off? I have a 20 meters.
Yeah. Well, let me just get a couple more meters here.
Okay. Yeah, Patty was remarking
like
about the Sea Star that was had all of its arms intact. We often don't see, or we often see Sea Stars that are missing arms. So this one is in really good shape.
I'm probably just gonna get this ship going ahead east.
Okay.
So shouldn't be anything out that way.
So still no wind.
Just come up maybe 10 meters, something like that.
So we're going to go ahead and have our little post dive, so all of you can head off. Some of you to bed.
There we go.
Hey everybody, it's Brian Kennedy. Who's still on the phone?
Bruce Mundy from IRC.
Hey Bruce, how's it going? Anybody else?
Good.
Harvey Bridge.
I know there's a couple people still in the event log, so they're probably listening in.
Yeah, we are
so
off
as Chris said, I don't think we need, or recommend we don't do a full-on post-dive call because we don't have very good data yet on the location we're going to. But first is, Chris, do you want to do any wrap-up for today's call? Anything you want to call out or summarize?
Yeah, so the dive was interesting in just the diversity of animals that we saw in this dive. As I mentioned, I wrote 2 pages of animal names here just for the dive summary report.
I need to stow
so we saw a lot of different things, but we didn't see a whole lot of.
Should I do that now, or should I do it in the water column?
One thing or several things. The density was lower than I was hoping for, but it was still a pretty interesting dive.
I'm not even turned yet. You want me to turn around? Okay.
I guess hope you enjoyed it. We got our 2 rocks, our 2 biologics, got some really beautiful close-ups and covered some ground. So all in all, a pretty successful dive.
Nice. Bruce, anything you want to add?
Okay.
Well, not
and
really.
Push out. Joy Locket pushed out a little bit and coming up.
Fishes, but it is very interesting to start explorations in this part of the ocean because as far as I know,
I'm
the
headed out then.
region here and over at Wake Atoll are basically totally unexplored for their deep sea fauna.
Yeah. Great. Okay. Well, I think since we don't have any data to look at, we will be in touch tomorrow, probably pretty late, because it's going to take us almost 25, 26 hours to get over to the site, and that'll give us 5 or 6 hours to map
the
Seamount.
if you can see it.
So we'll be in touch tomorrow with some more details, but most likely it'll be early morning our time, morning East Coast time, before we get any dive plans or anything out, since we don't have much data to go on. So we'll be in touch, and we'll talk to you guys all in 24 hours.
Out, so yeah, there we go.
Thank you very much.
Thanks, Rory. Have a good day.
Bye-bye.
I'll come up a little bit more.
Okay. Yep. We have 20 minutes.
Sounds good, but I'll be back on Monday.
Thanks, Bruce.
Have a good weekend, Bruce.